Welcome to Bedtime Biology, where science slows down and nature whispers you to sleep. Take time to find a comfortable position as you let the outside world fade away. Let it recede as you bring your attention to the present moment. Settle in, relax, and let's begin. Let your eyes close for a moment, and let the weight of the day settle quietly behind you. Picture a forest floor at dusk, the last soft light filtering down through a high canopy of leaves, catching on drifting motes of dust and the fine, damp mist rising gently from the ground. The air here smells of rich soil and old wood, of rain that fell days ago and never quite finished draining away. And scattered across a fallen log, pale and patient, a cluster of small mushrooms has quietly pushed itself up through the bark overnight, unhurried, exactly the way mushrooms have been doing in forests like this one for hundreds of millions of years. Somewhere nearby, an owl calls softly once, then settles into silence again. The last birdsong of the day has already faded, replaced by the low, steady hum of insects beginning their own evening chorus. Nothing here is in any hurry at all, and there's no reason you need to be either. We're going to spend this whole quiet journey together in the company of mushrooms and the far larger, far stranger organisms they belong to. Not to catalog them coldly, but simply to wander slowly through their world for a while, the way you might wander through an old, familiar forest at the very end of a long day, with nowhere else you need to be. There is nowhere else either of us needs to be right now. Let your breathing slow, settling into something as unhurried as the quiet, patient growth happening all around this small cluster of mushrooms tonight. Here's something worth sitting with gently before we go any further. That mushroom resting on the log isn't really the organism itself, not in the way we usually think of an individual living thing. It's closer to a piece of fruit, a temporary, above-ground structure produced by something vastly larger hidden beneath the surface, a sprawling, thread-like network called mycelium, spreading through the soil and the rotting wood in every direction, sometimes for great distances, entirely out of sight. The mushroom we can see is simply the part built to release spores into the world, the visible tip of something patient and enormous quietly living almost entirely in the dark. Mushrooms and the wider fungal kingdom they belong to aren't plants at all, despite how often they get lumped in with them. In fact, genetically speaking, fungi sit somewhat closer to animals than to plants, sharing certain deep evolutionary features that plants don't have at all. Their cell walls are built from a tough material called chitin, the very same substance that forms the hard outer shell of an insect or a crab, rather than the cellulose that gives a plant its structure. It's one of many small, quiet reminders that fungi have always followed their own particular path, refusing to fit neatly into the categories we build for the rest of the living world. That same close evolutionary connection to animals runs deeper still. Researchers comparing the genetic sequences of fungi, plants, and animals have found fungi and animals share a more recent common ancestor with one another than either group shares with plants, meaning the mushroom quietly growing on that same fallen log is, in a genuinely real evolutionary sense, a more distant cousin to the oak tree towering above it than it is to us. An association most people would never guess simply by looking at something so utterly unlike an animal in every outward appearance. Rather than producing their own food through sunlight the way a plant does, fungi feed instead by quietly breaking down whatever organic material surrounds them, releasing enzymes into dead leaves, fallen branches, and decaying wood, dissolving them slowly from the outside before absorbing the nutrients released. It's a patient, unglamorous process, and an absolutely essential one. Without fungi working steadily away in every forest floor and every patch of soil on this planet, dead trees and fallen leaves would simply pile up indefinitely, their nutrients locked away forever rather than return gently to the soil for the next generation of growth. Researchers estimate that fungi, working alongside bacteria and various small soil creatures, are responsible for breaking down the overwhelming majority of dead plant material produced across the world's forests every single year, an achievement so fundamental to how this planet's ecosystems function that without it, forests as we understand them simply couldn't exist in anything like their current form at all. It's easy to overlook something this quiet and this constant, precisely because it never once announces itself, simply continuing, patiently, in the background of nearly everything else happening in a forest at any given moment. Let your own breathing settle a little further now, slow and unhurried, the way this whole forest floor seems to breathe collectively, one long, patient exhale stretched out across an entire season. Somewhere beneath a forest in the state of Oregon, in the United States, a single fungal organism has been quietly spreading through the soil for what researchers estimate may be several thousand years, its mycelium now stretching across an area of forest covering nearly 4 square miles, making it, by some measures, the largest single living organism ever documented on this planet, larger than any whale, any redwood tree, anything else we might normally picture when we imagine an enormous, living thing. It belongs to a group of fungi known as honey fungus, and most of it remains completely invisible, threading quietly through root systems and buried wood, only occasionally sending up small clusters of honey-colored mushrooms to release its spores into the surrounding forest air. Researchers first began to suspect the true scale of this same organism through careful genetic sampling, testing tissue collected from mushrooms and infected roots scattered across a wide area, and finding, again and again, an identical genetic signature repeating itself across the entire span of forest, confirming that what looked, from the surface, like countless separate fungal colonies was in fact one single, continuous, genetically identical individual, patiently connected beneath the ground the whole time. That same discovery, published decades ago now, quietly reshaped how many biologists think about the very question of what counts as a single organism at all, since so much of our intuition about individuality comes from creatures with a clear, visible boundary, a skin, a shell, an obvious edge separating one living thing from everything around it, an intuition this same patient, sprawling fungus simply doesn't fit into it all. Picture that scale for a moment, if you can. An organism old enough to have been quietly spreading before many of the oldest trees in that same forest had even sprouted, still growing today, patient and entirely unhurried, at a pace so slow it becomes almost meaningless to measure in any unit of time we'd normally use for something alive. Let that same unhurried pace settle into your own body now too. There is nothing here that needs to happen quickly. Whatever tension you carried into this moment can simply soften, the way old wood softens slowly beneath a patient, spreading mycelium, one quiet season at a time. Beneath nearly every healthy forest floor, an entire hidden web of fungal threads connects individual trees together in ways scientists have only recently begun to properly understand. Fine fungal filaments wrap around and grow directly into the smallest root tips of a tree, forming a partnership called a mycorrhizal relationship, trading minerals and water drawn patiently from the surrounding soil in exchange for sugars the tree produces through its own leaves. This same fungal network often stretches on to connect with the roots of neighboring trees too, sometimes of an entirely different species altogether, weaving an entire forest into one loosely connected, underground web that researchers have taken to calling, a little poetically, the Wood Wide Web. Researchers estimate that the overwhelming majority of land plants alive today, somewhere well above 80% of all known species, depend on exactly this same kind of mycorrhizal partnership to survive, an association so fundamental and so widespread that it's genuinely difficult to find an ordinary, healthy forest, meadow, or garden anywhere that isn't, in some quiet way, held together by this same ancient, patient collaboration between root and fungal thread. Some studies suggest that through this same shared network, older, larger trees, sometimes affectionately called mother trees by the researchers who study them closely, may be able to direct extra nutrients towards smaller, struggling seedlings nearby, particularly their own offspring, using the fungal network as a kind of quiet, patient lifeline. Other researchers remain more cautious about how much genuine intention or awareness to read into this same behavior, noting that resources may simply flow toward wherever fungal and root pressure naturally draws them, without anything resembling deliberate care behind it at all. Either way, the underlying picture remains remarkable, an entire forest, quietly interconnected beneath the surface, far more thoroughly than anyone walking above the ground would ever guess simply from looking at the trees themselves. Researchers have also found that different tree species connected through the very same shared network don't always contribute or draw resources equally, some species acting more consistently as net givers across a season, quietly supporting neighboring trees of an entirely different species, while others draw more heavily from the shared network during their own particular periods of stress or vulnerability. That same shifting balance, some trees giving generously in one season and drawing more heavily in another, suggests a genuinely dynamic, ongoing relationship rather than any fixed, one-directional flow, an entire underground economy quietly rebalancing itself, season after season, according to whichever tree happens to need support most at any given moment. That same fungal network can also carry warning signals between trees, some experiments finding that a tree under attack from insects releases certain chemical signals into the shared mycelium, prompting nearby trees connected to that same network to begin producing their own defensive compounds slightly earlier than they otherwise would have, a quiet, chemical head start passed along through fungal threads long before any insect ever reaches them directly. Researchers running these same careful experiments typically compare an attacked tree's neighbors connected through shared mycelium against otherwise similar neighboring trees deliberately left unconnected, finding the connected trees consistently better prepared once the same insects eventually arrive, a difference measurable enough to rule out simple chance. It remains one of the more genuinely compelling pieces of evidence supporting the idea that this hidden fungal network carries real, functional information between trees, however cautious researchers remain about how much conscious intention to read into the process itself. Rest here a while, picturing that whole hidden, patient web spreading beneath the forest floor, connecting root to root, tree to tree, an entire community bound together by something most of us will spend our whole lives walking directly above without ever once realizing it's there. As true darkness settles in now, something else quietly reveals itself among the scattered, decaying logs of this same forest floor. Here and there, a faint, pale green glow begins to emerge from patches of old, rotting wood, soft enough that you'd need your eyes fully adjusted to the dark to notice it at all. This is foxfire, caused by certain species of bioluminescent fungi quietly living within the decaying wood, producing their own soft light through much the same basic chemical process fireflies and certain deep-sea creatures rely on. One particular mushroom responsible for this same eerie glow, sometimes called the jack-o'-lantern mushroom for its warm orange color and gently luminous gills, grows in dense clusters at the base of trees and stumps across parts of North America, Europe, and Asia. Its gills alone produce enough light that early photographs of the phenomenon required exposures lasting several hours simply to capture the glow clearly, since to the human eye alone, the light is faint enough that many people walk straight past it without ever noticing anything unusual at all. Researchers measuring the exact chemistry behind this same glow have found it relies on the very same basic luciferin and luciferase reaction responsible for a firefly's own light, a striking example of entirely separate branches of the tree of life, fungi, and insects, arriving independently at almost the very same chemical solution for producing their own soft, living light, evolved millions of years apart from one another and yet converging, once again, on remarkably similar underlying machinery. Historically, miners and soldiers in the field sometimes relied on pieces of this same glowing, decaying wood as an improvised, if very faint, source of light, tucking a piece into a lantern or simply carrying it directly, since an open flame in certain settings, particularly deep underground where flammable gases might be present, posed a considerably greater danger than the mushroom's own soft, cool glow ever could. Soldiers during the First World War, stationed in damp, wooded trenches across parts of Europe, are documented to have occasionally gathered pieces of glowing wood specifically to help mark safe pathways in the darkness, a small, soft point of light considerably less likely to draw unwanted enemy attention than any conventional lamp or torch would have. It's a quietly remarkable thought, that in the midst of one of history's most difficult periods, people found a small measure of practical help in exactly the same soft, patient glow we've paused to admire together tonight. Researchers still don't fully agree on exactly why certain fungi evolved this same soft bioluminescence in the first place. Some suggest the glow may help attract nocturnal insects toward the mushroom, insects that then carry its spores away to new locations after brushing past the glowing gills. Others suggest the light might simply be an incidental byproduct of ordinary fungal metabolism, serving no particular purpose of its own at all, a beautiful accident rather than any deliberate strategy. Let your own eyes rest gently now too, however curious you might be about that same soft, glowing wood. There's nothing you need to see clearly tonight. The glow will still be there, quiet and patient, whether your own eyes stay open to notice it or close instead, resting, the way the rest of this whole forest is slowly settling into its own night. Let's drift now toward a stretch of forest where birch and pine grow close together, their roots tangled quietly beneath a carpet of fallen needles and old leaves. Here, rising up through the moss, a mushroom instantly recognizable even to people who know almost nothing else about fungi pushes its way into view, a bright red cap scattered with small, white, flaky spots, exactly the image so many childhood storybooks borrowed for their own imagined toadstools. This is the fly agaric, and that same striking red and white coloring serves as a clear, honest warning. Her flesh contains compounds that are toxic if eaten, capable of causing intense nausea, disorientation, and in rare cases, more serious harm, though fatalities remain genuinely uncommon. In certain cultures across Siberia and parts of northern Europe, this same mushroom holds a much older, more complicated history, used carefully in small, prepared quantities within traditional shamanic practices for its unusual psychoactive effects. Practices considerably older than most of the folklore that later borrowed her image for entirely different purposes. The particular compounds responsible for her effects, called ibotenic acid and muscimol, behave quite differently from the toxins found in the death cap we'll meet shortly, working instead on the nervous system rather than the liver, and interestingly, traditional preparation across several of these same cultures involved careful drying of the mushroom beforehand, a process that measurably converts a portion of the more harsh ibotenic acid into the somewhat gentler muscimol, an old, practical piece of chemistry worked out through patient, careful trial long before anyone could have explained the underlying reaction in any scientific terms at all. Some historians have drawn an intriguing, if debated, connection between the fly agaric and more familiar modern imagery, pointing to old Siberian traditions involving reindeer, red and white coloring, and midwinter gift-giving rituals as a possible thread running through to the modern image of a certain red-suited, white-trimmed figure and his own reindeer, delivering gifts each winter. Whether that connection is genuine history or simply a charming coincidence remains an open question among folklorists, though it's the kind of detail that tends to stick pleasantly in the mind regardless. The fly agaric also depends entirely on a partnership with nearby trees to survive at all, her mycelium wrapping around the roots of birch, pine, and spruce in exactly the same mycorrhizal relationship we discussed a little earlier tonight, trading minerals for sugar, an entire hidden economy of exchange happening quietly underground every time you notice her bright, familiar cap rising up through a patch of moss. Let your own body settle a little heavier now, wherever you happen to be resting. There's nothing urgent in this next part of the story either, only the same slow, unhurried pace this whole forest has kept all night long. Not every mushroom announces its danger quite so plainly as the fly agaric does. Deeper in this same forest, tucked quietly beneath a cluster of oak leaves, a small, pale, unassuming mushroom grows, easily mistaken by an inexperienced forager for something considerably safer. This is the death cap, and despite her calm, ordinary appearance, she remains responsible for the overwhelming majority of fatal mushroom poisonings recorded anywhere in the world. Her danger lies in a group of compounds called amatoxins, which quietly and irreversibly damage the liver, often with a delay of many hours between the moment she's eaten and the first serious symptoms appearing, a delay long enough that by the time real trouble becomes obvious, significant harm has frequently already been done. Some historians have speculated that the Roman Emperor Claudius may have been deliberately poisoned using a dish involving death cap mushrooms, though the historical evidence for that particular theory remains genuinely uncertain, led over by centuries of political rumor and retelling. What makes amatoxins particularly dangerous, researchers studying the compound have found, is the way they interfere directly with a cell's own ability to manufacture new proteins, a fundamental process every cell in the body depends on constantly, and the liver, tasked with filtering and processing nearly everything absorbed into the bloodstream, ends up absorbing a disproportionate share of the toxin as a direct consequence of simply doing its own ordinary job. A single death cap, researchers estimate, contains enough amatoxin to seriously endanger an adult, and unlike many other toxins, the compound remains entirely unaffected by cooking, meaning no amount of careful preparation can ever render this particular mushroom safe to eat. A close relative, aptly named the destroying angel, carries almost identical danger inside a pure, unmarked white cap, easily mistaken by the unprepared for any number of ordinary, edible mushrooms sharing the same general shape. Both species serve as a quiet, serious reminder of something experienced foragers repeat constantly to anyone just beginning to learn. Appearance alone can never be trusted to reveal whether a wild mushroom is safe, and a single mistaken identification can carry consequences considerably more serious than an unpleasant evening. Let that same caution rest quietly in the back of your mind, without any tension attached to it. You don't need to remember every detail right now. Simply let the story continue, unhurried, exactly as it wants to. Not every mushroom carries danger, of course. Considerably further along this same forest path, growing in overlapping, fan-shaped clusters along the side of a dead tree, a mushroom marked with concentric bands of brown, tan, and cream catches what little light remains in the evening sky. This is turkey tail, named for its close resemblance to the fan tail feathers of the bird it borrows its name from, and one of the most extensively studied mushrooms anywhere in the world for its potential medicinal properties. Compounds extracted from turkey tail have been studied for decades, particularly in Japan, where a purified extract derived from the mushroom has been used for years alongside conventional cancer treatment, believed to help support the body's own immune response during recovery. Research into exactly how effective this same treatment truly is remains ongoing and, in parts of the world outside Japan, is treated with somewhat more caution, though the sheer volume of careful, patient research already conducted makes turkey tail one of the better documented medicinal mushrooms studied by modern science. Beyond her medicinal reputation, turkey tail plays a genuinely important ecological role too, an efficient, widespread decomposer of dead hardwood found across nearly every temperate forest in the northern hemisphere. Her fanned, banded clusters among the most commonly encountered bracket fungi anywhere, a familiar, patient presence quietly breaking down fallen branches long before most passing walkers ever think to identify her by name. Not far away, growing directly from the base of a maple tree, a mushroom with a glossy, reddish-brown, almost lacquered cap catches the last of the light beautifully. This is reishi, treasured across East Asian traditional medicine for many centuries under names that translate roughly to something like the mushroom of immortality, valued traditionally for supporting calm, restful sleep and general vitality. Modern research continues carefully examining reishi's compounds for their effects on immune function and stress, a slow, patient scientific echo of an appreciation that traditional healers have held for this same mushroom across countless generations already. Elsewhere, growing not from wood at all but directly from the base of certain hardwood trees, an unusual mushroom with long, cascading white spines instead of the more familiar gills or pores catches the eye, resembling, at a glance, a soft, shaggy waterfall frozen mid-cascade. This is lion's mane, and in recent years, researchers have taken particular interest in compounds found within it that appear, at least in laboratory and animal studies so far, to encourage the growth of nerve tissue, a genuinely exciting area of ongoing research into whether this same mushroom might one day support cognitive health and nerve regeneration in people more directly. The research remains in its earliest stages, careful and unhurried exactly the way good science should be. But the early findings have been intriguing enough to draw considerable continued attention from researchers around the world. Traditional use of lion's mane across parts of China and Japan stretches back centuries, prized historically both as a genuinely delicious ingredient, her texture when cooked often compared to crab or lobster, and as a tonic believed to support general vitality and clear thinking, long before any modern laboratory ever isolated the specific compounds now drawing careful scientific attention. It's one more example of traditional, patient observation arriving early at something modern research is only now beginning to properly understand and explain in careful, precise detail. High on the trunk of a birch tree, a dark, cracked, almost charcoal-like growth clings to the bark, looking, at first glance, more like burnt wood than any kind of mushroom at all. This is chaga, and unlike most of the fungi we've met so far tonight, what you're actually looking at is a hardened mass called a sclerotium, a dense, protective structure the fungus builds up slowly over many years, sometimes decades, before it ever produces anything resembling a conventional mushroom at all. Harvested carefully and traditionally brewed into a strong, earthy tea across parts of Russia, Scandinavia, and elsewhere, chaga has been valued for generations for its antioxidant properties, a slow, patient fungal growth transformed, just as slowly and patiently, into a warm drink meant to be sipped unhurried, exactly the way it was grown. Traditional harvesters take particular care to leave a portion of the chaga growth intact on the tree whenever they harvest, understanding through generations of accumulated experience that a fully removed growth may take considerably longer, sometimes many additional years, to properly regenerate, if it manages to at all. That same careful, patient restraint echoes precisely the same sustainable harvesting principles we've returned to again and again throughout tonight's journey, taking only what's genuinely needed while leaving enough behind for this same patient, slow-growing partner to continue thriving for many seasons still to come. And finally, cultivated carefully on logs and specially prepared beds across much of East Asia for well over 1,000 years, a mushroom with a broad, umbrella-shaped brown cap and pale gills has become one of the most widely eaten mushrooms anywhere on the planet. This is shiitake, and beyond her rich, savory flavor, treasured in kitchens worldwide, she carries her own long, careful history of cultivation, early farmers learning, through patient trial across many generations, exactly which logs, exactly which conditions, would coax her mycelium into producing a reliable, abundant harvest. Long before anyone fully understood the biology explaining why those particular methods worked so well. Rest here a while amid all these different mushrooms, the beautiful and the dangerous, the ancient and the carefully cultivated. There's nothing more you need to sort or remember right now. Just let this same unhurried forest continue quietly around you, exactly as it has this whole time. Let's continue drifting through this same quiet forest, toward an open, grassy clearing where the trees briefly part and the sky opens up overhead. Here, scattered across the damp grass, several round, pale, ball-shaped growths rest quietly against the earth, some no larger than a golf ball, others considerably larger, closer to the size of a small pumpkin. These are puffballs, and unlike nearly every mushroom we've met so far tonight, they carry no cap, no gills, no stem in the way we usually picture a mushroom's shape. Instead, their entire fruiting body forms one solid, rounded mass, packed densely inside with countless trillions of spores, maturing slowly over the course of a season until, eventually, the outer skin dries out completely and finally splits open. The lightest touch, even a single heavy raindrop landing directly on the surface, is enough to send an entire visible cloud of fine, dusty spores puffing out into the surrounding air, carried onward by whatever breeze happens to be passing through at that exact moment. The giant puffball, true to her name, can grow to genuinely enormous size, with some recorded specimens exceeding a meter across and weighing more than 20 kilograms, an almost unbelievable size for something so closely related to the small, familiar mushrooms scattered across the forest floor nearby. When young, before that same soft, white interior has matured into the countless dry spores it will eventually release, a puffball is considered a genuine culinary delicacy in several traditions, sliced and cooked while still firm and mild, long before the spore-filled maturity that gives the mushroom her memorable name. Foragers checking whether a puffball is still young and safe enough to eat typically slice one open first, looking for flesh that remains pure, uniform white all the way through. Any hint of yellowing, or the faint outline of a developing cap and stem visible within the flesh, signals either an aging puffball no longer suitable for the table, or, more importantly, one of a small number of unrelated, genuinely dangerous young mushrooms that can occasionally resemble a puffball at a very early stage of their own development. One more example of why the same careful, patient identification we discussed earlier tonight always matters, regardless of how straightforward a particular mushroom might initially seem. Researchers studying puffball spore counts have calculated that a single large specimen may release something in the range of 7 trillion individual spores across her whole lifetime, a number so large it becomes almost meaningless to picture directly. And yet, out of that entire trillion-strong cloud, only an infinitesimally small fraction will ever actually land somewhere suitable enough to grow into a new colony of mycelium. It's a strategy built entirely around abundance rather than precision, trusting sheer overwhelming numbers to ensure that at least a few spores, somewhere, eventually find themselves in exactly the right patch of soil. To put that same trillion spore figure in some kind of perspective, if every single spore released by just one large puffball successfully grew into a new colony, the resulting mycelium would, within only a handful of generations, cover an area of ground many times larger than the entire surface of the planet. That such abundance produces, in practice, only a modest scattering of new colonies each season says everything about just how narrow the odds of any single spore's success actually are, and just how completely this whole patient strategy depends on numbers alone to make up for that same narrow, uncertain chance. Let your own breathing settle into that same unhurried abundance for a moment, needing no precision at all right now, simply trusting that rest, like those countless drifting spores, will eventually settle exactly where it needs to. Not far from this same clearing, tucked half hidden in a patch of damp leaf litter, an entirely different fungal strategy has taken root, one relying not on quiet patience at all, but on speed and a genuinely unusual smell. Rising up from a soft, egg-shaped structure buried in the soil, a tall, slender stalk has pushed rapidly upward, capped at the top with a dark, slimy substance that gives off an odor unmistakably similar to rotting meat. This is a stinkhorn, and that same unpleasant smell serves a deliberate, effective purpose. Flies and other carrion-loving insects, drawn in eagerly by what their own senses interpret as the promise of decaying flesh, land directly on the sticky, spore-laden cap, picking up countless spores on their legs and bodies before flying off to investigate the next promising, foul-smelling scent they happen to encounter, unknowingly carrying the stinkhorn's own spores to an entirely new location in the process. Researchers analyzing the precise chemical compounds responsible for a stinkhorn's smell have found her scent genuinely, remarkably close in composition to the chemical signature of actual decaying flesh, evolved with such fine-tuned precision that the flies she attracts show no apparent hesitation at all, landing and feeding exactly as they would on genuine carrion, entirely unaware they've been thoroughly, elegantly deceived by nothing more than a patient, stationary mushroom. Stinkhorns are also remarkable for the sheer speed of their growth, the entire visible stalk capable of pushing up from the ground and reaching its full height in a matter of just a few hours, one of the fastest rates of growth recorded anywhere in the plant or fungal kingdoms. A patch of forest floor that showed no sign of a stinkhorn at breakfast might, by the time evening arrives, already carry several fully grown specimens, standing tall and pungent, patiently waiting for the right insect to arrive and complete their particular, unusual strategy for spreading spores across the forest. Let's drift now toward a shaded, damp log nearby, where something even stranger moves slowly across the surface of the bark, something that isn't actually a fungus at all, despite how often the two get mentioned in the very same breath. A soft, yellow, almost web-like mass creeps steadily forward across the wood, visibly shifting its own shape over the course of just a few hours, an organism called a slime mold. Slime molds occupy their own strange, fascinating branch of the tree of life, neither fungus, nor plant, nor animal in any straightforward sense, though they share certain habits with fungi that have long caused the two to be casually grouped together. One particular species, studied closely by researchers for decades now, exists as a single enormous cell containing many thousands of individual nuclei, spreading itself out across considerable distances in search of the bacteria and organic debris it feeds on. What makes this same slime mold so remarkable to researchers is a genuine capacity for solving problems, despite having no brain, no nervous system, no centralized control of any kind at all. In one especially well-known experiment, researchers placed a slime mold inside a maze with oat flakes positioned at two separate exits, mimicking the layout of a real city's rail network. Within a day, the slime mold had reorganized its own spreading body into a pattern of connections closely mirroring the actual, carefully engineered rail network the maze was designed to represent, effectively solving a genuine urban planning problem through nothing more than its own patient, exploratory growth. Researchers have since used slime molds to model efficient network design in several other real-world contexts too, always arriving, through nothing more sophisticated than patient trial and gradual reinforcement of whatever pathway happens to work best, at solutions remarkably close to what considerably more calculated human engineering would eventually produce on its own. It stands as a genuinely humbling reminder that intelligence, in some very real sense, doesn't always require anything resembling a brain at all, sometimes emerging instead from nothing more than patient, distributed trial and error, repeated quietly over and over until the best answer simply, gradually, reveals itself. Researchers have also found slime molds capable of a genuinely surprising form of memory, despite having no nervous system whatsoever to store information in any way we'd normally recognize. After exploring and rejecting a particular pathway once, a slime mold will often avoid retracing that same unproductive route on a later exploration, apparently leaving behind some kind of chemical trail marking where it has already searched, a simple but genuinely effective external memory laid down directly in its own path of movement, allowing it to explore new territory more efficiently rather than wastefully repeating ground already covered. Rest here a while longer, watching this same slow, patient creature continue its unhurried spread across the old log, solving its own quiet problems one careful, exploratory inch at a time, exactly the way the rest of this whole forest continues solving its own problems too, slowly, patiently, without any need to rush toward an answer at all. Let's move now beneath the surface entirely, down into the soil itself, where an entirely different kind of fungus has chosen to grow its fruiting body completely underground, never once breaking through into open air the way every mushroom we've followed so far tonight has done. Tangled among the roots of an old oak tree, a small, irregular, knobbly growth rests hidden beneath the soil, invisible from above, entirely undetectable except by scent alone. This is a truffle, and her entire strategy for spreading spores depends on smell rather than sight, releasing a rich, powerful aroma directly into the surrounding soil, an aroma capable of drawing in animals from a considerable distance despite being completely buried and completely invisible. Wild boar, in particular, have long been drawn instinctively to that same rich scent, digging down to unearth and eat the truffle, spores passing safely through the animal's digestive system and eventually being deposited, along with a convenient bit of natural fertilizer, somewhere entirely new. People, too, have learned to take advantage of that same powerful scent, though these days considerably more often with the help of specially trained dogs than the traditional pigs once used across parts of France and Italy, dogs proving both easier to work with and considerably less inclined to simply eat the valuable truffle themselves the moment it's found. A skilled truffle dog can detect the scent of a mature truffle buried as much as a meter beneath the surface, an extraordinary feat of scent detection built on months of careful, patient training between a dog and her human handler. That same training typically begins when a dog is still quite young, gradually associating the specific scent of truffle with a reward, long before she's ever taken into an actual forest to search in earnest. Experienced trainers describe the process as requiring considerable patience on both sides, dog and handler alike gradually learning to read one another's subtle signals, a slight change in pace, a particular way of pausing and sniffing more intently, until the pair can work together through an unfamiliar patch of forest with genuine, practiced confidence, each one trusting the other completely by the time a serious harvest season finally arrives. Certain truffle varieties, particularly the white truffles found in specific regions of Italy, command remarkably high prices, driven by a combination of genuine rarity, a distinctive flavor difficult to replicate through any other ingredient, and the simple fact that truffles have never been successfully, reliably cultivated at scale the way shiitake or other mushrooms have. Truffle cultivation does exist, careful orchards planted with young oak or hazel saplings deliberately inoculated with truffle spores, but even then, a successful harvest remains considerably less certain and considerably more dependent on exactly the right soil, climate, and simple good fortune than most other cultivated crops ever need to worry about. Let your own body rest a little deeper now too, tucked away and hidden, the way this same truffle rests quietly beneath the soil, needing no attention at all from the world above in order to continue its own patient, unhurried existence. Let's rise back up now, out of the soil and into a warm, humid forest considerably further from here, somewhere in the tropics, where an entirely different, considerably stranger fungal strategy plays out among the insects moving across the forest floor and up into the surrounding trees. High on the underside of a single leaf, a small ant clings motionlessly in place, jaws clamped firmly around the leaf's central vein, already dead, though her body remains standing exactly as if she were still alive. Rising up from the back of her head, a single, slender stalk extends outward, tipped with a small, dark structure that will soon release its own spores down onto the forest floor below, directly into the path of other ants from the very same colony passing beneath. This is cordyceps, sometimes called, a little dramatically but not inaccurately, the zombie fungus, a genuinely remarkable parasite capable of infecting a specific species of ant, growing quietly through her body while somehow influencing her behavior along the way, guiding her, in her final hours, to climb to a very particular height and position, biting down permanently onto a leaf vein in a location that researchers have found offers the fungus its own ideal conditions of temperature and humidity for eventually releasing spores most effectively. Different cordyceps species have evolved to target different specific insect hosts, each one exquisitely adapted to manipulate its own particular prey with remarkable precision, a testament to how thoroughly and how patiently evolution has refined this same strange, parasitic relationship across countless generations. Researchers studying exactly how the fungus manages to influence its host's behavior so precisely, without any direct access to the ant's brain itself, continue to find the mechanism genuinely difficult to fully untangle, the fungus seeming to influence surrounding muscle tissue directly instead, effectively hijacking the ant's own body from the outside in, rather than issuing commands from any central control point within the brain at all. Some ant colonies, researchers have found, have developed their own careful countermeasures against this same patient parasite, worker ants apparently capable of detecting the earliest, subtle signs of infection in a nestmate and physically removing her from the colony before the fungus has any chance to mature and release its own spores nearby, an entire quiet, ongoing evolutionary standoff playing out between parasite and colony, each side patiently refining its own strategy across countless generations in response to the other. One particular cordyceps species, found growing on caterpillars high in the mountains of Tibet and Nepal, has been prized in traditional Chinese medicine for centuries, harvested carefully each spring by hand across remote, high-altitude meadows, and valued as highly, in certain markets, as gold itself by weight. That same considerable value has, in recent decades, transformed harvesting this particular fungus into a significant seasonal economic activity for entire mountain communities, an old, traditional practice now intersecting with a very modern, considerable global demand. Rest here a while amid this same strange, patient parasite, neither entirely comfortable nor entirely unsettling, simply another quiet example of just how many different, careful strategies this whole fungal kingdom has developed over its own immense span of time on this planet, each one shaped patiently across millions of years into exactly the particular, precise form it needed to become. Let's travel now toward a stretch of bare, exposed rock at the edge of the forest, where almost nothing else seems willing to grow at all. And yet, spread flat and patient across the stone's uneven surface, a thin, papery crust of pale green and gray has quietly established itself, seemingly content with nothing more than bare mineral and whatever moisture happens to drift by on the passing air. This is lichen, and it isn't a single organism at all, but rather a patient, long-term partnership between a fungus and a photosynthetic partner, usually a simple alga or a cyanobacterium, living together so closely and so completely that the combined result behaves, in every practical sense, like one single, unified organism. The fungal partner provides physical structure and protection from drying out, while the photosynthetic partner quietly manufactures food from sunlight, sharing the resulting sugars generously with its fungal host in return. That same careful partnership allows lichen to survive in places that would overwhelm almost any other form of life: bare rock faces, the frozen ground of the high Arctic, exposed mountain peaks battered constantly by wind and cold. Some lichens have even been sent deliberately into open space aboard research satellites, exposed directly to the vacuum, the radiation, and the extreme temperature swings of low orbit, and remarkably, many samples returned to Earth still very much alive, having simply endured conditions that would have killed almost any other living thing within moments. Researchers running these same space exposure experiments found the lichen's fungal partner appeared to play a particularly important protective role, its own outer tissue shielding the more vulnerable photosynthetic partner tucked safely within, much the way it shields that same partner from ordinary drying and temperature extremes here on Earth, simply extended to a considerably more extreme setting than anyone had previously tested. The experiment offered genuine, careful evidence that this ancient, patient partnership might be resilient enough to survive conditions found on other worlds too. A small, quiet possibility researchers studying the potential for life beyond Earth continue to find genuinely worth taking seriously. Lichens also grow at a pace so slow it becomes genuinely difficult to comprehend clearly. Certain species have been measured growing less than a single centimeter across an entire century, meaning some of the larger lichen patches found on ancient, undisturbed rock faces may be many thousands of years old, quietly older than most of recorded human history, still patiently spreading, millimeter by careful millimeter, across the very same stretch of stone. Let your own body settle into that same patient stillness for a moment. There's no need to grow, or change, or accomplish anything at all right now. Simply resting, exactly where you are, is more than enough. Because lichens draw so much of what they need directly from the surrounding air, they've also become remarkably useful to researchers as a kind of natural, living indicator of air quality. Certain especially sensitive lichen species will simply disappear entirely from an area once air pollution reaches a certain threshold, unable to tolerate the change, while more tolerant species continue on largely unaffected. Scientists studying air quality across cities and industrial regions have, for decades now, used the presence, absence, and particular variety of lichen growing on nearby trees and rock as a simple, elegant, and remarkably reliable gauge of how clean or polluted the surrounding air actually happens to be, an entire quiet early warning system built from nothing more than careful, patient observation of something most people walk straight past without a second glance. Let's drift now toward somewhere considerably warmer and closer to home, toward a quiet kitchen where a small bowl of dough sits resting on a counter, slowly, steadily rising beneath a light cloth. The organism responsible for that same gentle, patient rise is a fungus too, though one so small that a single individual cell remains entirely invisible without the help of a microscope. This is yeast, and unlike every other fungus we've followed so far tonight, she exists not as a network of branching threads, but as countless individual, single-celled organisms, each one capable of quietly converting sugar into carbon dioxide and alcohol through a patient chemical process called fermentation. In this same bowl of rising dough, that same carbon dioxide is what's doing the actual work, countless tiny bubbles forming slowly throughout the dough, causing it to expand gently, hour by unhurried hour, into the soft, airy bread it will eventually become. That same fermentation process, when carried out instead in a vat of grape juice or malted grain rather than bread dough, produces something rather different. Wine and beer, two of the oldest deliberately cultivated products in the whole of human history, refined patiently across thousands of years long before anyone fully understood the tiny, single-celled organism responsible for the transformation at all. Ancient bakers and brewers worked entirely by careful, repeated observation, noticing which particular batch of dough or which particular vat of liquid behaved most reliably, and simply continuing to use a small portion of that same successful batch to start the next one, unknowingly cultivating and preserving a specific, reliable strain of yeast across countless careful generations of practice. Beyond the kitchen, this same humble yeast has also become one of the single most important organisms in all of modern biological research, its relatively simple, well-understood genetics making it an ideal subject for studying the fundamental machinery shared by nearly every living cell, including our own. Countless discoveries about how cells divide, repair damage, and regulate their own genetic activity were first carefully worked out studying nothing more exotic than ordinary baker's yeast, patiently cultured in laboratories around the world, the same humble organism responsible for your morning toast quietly helping researchers unlock some of biology's deepest, most fundamental secrets. Yeast became the very first eukaryotic organism, meaning an organism whose cells carry a properly enclosed nucleus the way ours do, to have its entire genetic code fully mapped out, an achievement completed considerably earlier than the mapping of the human genome that would follow years later. Researchers chose yeast specifically because so much of the basic cellular machinery it relies on turns out to be remarkably similar to our own, despite the enormous outward difference between a single-celled fungus and a complex, multicellular person, a quiet reminder of just how deeply, at the most fundamental cellular level, so much of life on this planet remains genuinely, patiently connected. Rest here a while amid the quiet, patient rise of that same simple dough, and the countless invisible cells working steadily away inside it, needing nothing more from you right now than the same soft, unhurried patience they've already been demonstrating this entire time. Let's return now to the wider forest, and consider for a moment just how many different, careful strategies fungi have developed simply to spread their spores out into the world, each one shaped patiently by whatever particular environment that fungus happens to call home. We've already met several of these strategies tonight without quite naming them so directly. The puffball, releasing an entire visible cloud with nothing more than a single raindrop's gentle pressure. The stinkhorn, luring flies in with the promise of a meal that was never really there at all. But countless other approaches exist too, each one just as patient, just as carefully refined. Certain mushrooms, including many of the more familiar, gilled varieties scattered across almost any forest floor, rely on nothing more than gravity and the gentlest passing breeze, releasing spores continuously from thin, papery gills arranged beneath the cap, each spore drifting downward before catching whatever faint air current happens to be moving through the surrounding forest at exactly that moment. A single gilled mushroom may release many millions of spores across the course of just a few days, an entire quiet, continuous rain of microscopic potential drifting invisibly through the air around us far more often than most of us would ever think to imagine. Mycologists studying wild mushrooms have long relied on a simple, elegant technique to help identify a particular species, called a spore print. Resting a mushroom cap, gills facing downward, on a plain sheet of paper overnight allows the mushroom to quietly release its own spores directly onto the page below, forming a delicate, detailed pattern that reveals both the exact arrangement of the gills above and the true color of the spores themselves, a color that often differs meaningfully from the color of the cap or gills to the naked eye, and one that provides a genuinely important clue for correctly identifying a mushroom whose safety might otherwise remain uncertain. Spore color across the fungal kingdom ranges remarkably widely, from pure white through cream, pink, deep chocolate brown, rich purple-black, and even certain shades of yellow-green, and professional mycologists rely on this same careful color distinction as one of several essential clues, alongside the shape of the cap, the texture of the stem, and the particular smell a mushroom gives off, when working to correctly place an unfamiliar specimen within its proper family. A single spore print, prepared patiently overnight and examined carefully the following morning, can often settle in a matter of seconds a question that might otherwise take considerably longer to resolve through visual inspection alone. Some fungi have developed considerably more forceful methods of spore dispersal too. Certain species build up internal pressure slowly within a specialized structure, before finally releasing their spores in a genuinely explosive burst, launching them outward at considerable speed relative to their own microscopic size, sometimes described by researchers as among the fastest accelerations recorded anywhere in the natural world, achieved by an organism with no muscles, no nervous system, nothing beyond patient. Accumulated pressure finally released all at once. Other fungi depend entirely on animals to carry their spores onward, not through any deliberate law like the stinkhorn's rotting smell, but simply by being eaten directly. Certain mushrooms growing on animal dung have evolved spores that pass safely and completely unharmed through an animal's entire digestive system, emerging later in a fresh, new pile of dung considerably better suited to their own growth than the spot where they started out. Some of these same spores, researchers have found, actually require passing through an animal's digestive tract before they're able to germinate successfully at all, an entire life cycle patiently built around an unglamorous, if remarkably reliable, journey. Let your own thoughts drift now too, as loosely and unhurried as any one of these countless spores, carried gently forward without needing to control exactly where they land or precisely what happens next. Fungi's remarkable ability to break down and transform difficult, resistant material hasn't gone unnoticed by researchers looking for genuinely practical solutions to some of our more modern, human-made problems either. Certain species of fungi have been found capable of slowly breaking down plastic, and material that would otherwise persist essentially unchanged in the environment for centuries, using the very same patient enzymatic processes that allow other fungi to break down tough, resistant wooden bark. Researchers working in a field called mycoremediation have experimented with using specific fungi to help clean up land contaminated by oil spills too. The fungus's own natural enzymes slowly breaking down petroleum compounds into simpler, less harmful substances over the course of weeks and months, a slow, patient, biological alternative to some of the more aggressive chemical treatments traditionally used for the very same purpose. Early pilot projects testing this same approach on contaminated soil have shown genuinely promising results. Mycelium spreading steadily through polluted ground and leaving noticeably cleaner soil behind once its patient work is finally done. One especially notable pilot project involved contaminated soil left behind after a significant diesel spill. Researchers inoculating the affected ground with a specific oyster mushroom strain known for its efficient enzymatic activity, and returning months later to find not only measurably reduced contamination levels, but the soil itself once again capable of supporting healthy plant growth. Insects and other small creatures gradually returning to a patch of ground that had, only months earlier, been essentially lifeless. It stands as one of the more hopeful, tangible demonstrations of exactly how much quiet, patient help this ancient kingdom is capable of offering, once people take the careful time to properly understand and direct its own natural talents toward a genuinely useful purpose. Some researchers have also begun exploring whether certain mushroom mycelium might be grown deliberately into sturdy, lightweight materials entirely on its own, dense mats of fungal thread pressed and dried into shapes strong enough to replace traditional packaging material, or even certain kinds of building insulation, and entirely renewable, biodegradable material grown patiently over the course of just a few days rather than manufactured through any energy-intensive industrial process at all. Rest here for a while, considering everything this quiet, patient kingdom has already offered the world, freely and without complaint, long before anyone ever thought to ask for its help directly. There's nothing more you need to understand about any of it right now. Simply let the thought settle, gently, the way everything else tonight has settled too. Let's wander now toward a slightly different stretch of forest, where the ground beneath a stand of old ash and elm trees is scattered with small, honeycombed, cone-shaped mushrooms rising directly from the soil each spring. These are morels, among the most highly prized wild mushrooms foraged anywhere in the world, their distinctive, deeply pitted caps making them relatively easy to identify with confidence compared to many of their more ambiguous relatives. Morels have never been reliably cultivated at any real commercial scale, despite considerable effort by researchers and mushroom farmers alike over many decades. Something about the particular combination of soil conditions, temperature, and often the presence of specific dying or recently dead trees nearby remains difficult to reproduce artificially with any real consistency, meaning nearly every morel found in a kitchen anywhere in the world was patiently foraged by hand from the wild, exactly the way it would have been centuries ago. Foragers often return, year after year, to the very same patch of forest where they once found a good morel harvest, since the mushroom shows a genuine tendency to reappear in the same general area across successive seasons, drawing on the same patient, established mycelium quietly persisting underground the whole time. Morels also show a particular, well-documented tendency to fruit abundantly in areas affected by recent forest fires, their mycelium apparently responding to exactly the same kind of soil disturbance and chemical change we discussed earlier tonight regarding pyrophilous fungi, though morels themselves aren't considered true fire specialists in quite the same strict sense. Experienced foragers across parts of the western United States have learned to watch burned areas particularly closely the following spring, sometimes finding remarkably abundant harvests in a landscape that, only months earlier, looked entirely too devastated to offer anything at all. Not far away, growing in bright, golden-orange clusters directly from the soil beneath a stand of oak trees, chanterelles offer foragers a considerably more reliable, if still genuinely challenging, harvest. Their delicate, fruity aroma, often compared to apricots, and their gently ruffled, false gills, really just shallow ridges rather than true, blade-like gills, help distinguish them from certain toxic lookalikes, though as always, experienced foragers stress the importance of complete certainty before ever eating anything found growing wild. Chanterelles, like so many of the mushrooms we've already discussed tonight, depend entirely on a mycorrhizal partnership with the trees growing nearby, meaning they too have resisted reliable large-scale cultivation, remaining instead a genuinely wild harvest, gathered carefully by hand from forests across multiple continents, patiently, one careful basket at a time. Let your own body settle a little further now, resting easy, the way a forager might rest after a long, satisfying afternoon spent slowly working through a familiar patch of forest, needing nothing more right now than the quiet satisfaction of simply having been there, present, unhurried. Growing instead directly from the side of a dying or fallen tree, in soft, overlapping clusters resembling pale, fan-shaped shells, oyster mushrooms represent one of the more successful stories in modern mushroom cultivation, grown commercially now across the world on everything from prepared straw to spent coffee grounds, an efficient, adaptable fungus willing to make a home in almost any suitable organic material offered to it. Their mycelium works through whatever substrate they're growing on with genuine efficiency, converting agricultural waste that might otherwise simply be discarded into a fresh, edible harvest within just a few weeks. Oyster mushrooms carry one further, considerably more unusual trait too. Certain related species have been found capable of trapping and consuming microscopic worms called nematodes, the fungus producing small, sticky structures along its own mycelium that immobilize any nematode unfortunate enough to brush against them, after which the fungus slowly digests its unexpected catch directly, supplementing its usual diet of decaying wood with a small, occasional source of additional nutrients. A startling reminder that even a mushroom we think of primarily as food can, itself, occasionally turn predator. Researchers examining this same predatory behavior under a microscope have found the entire process unfolds with genuine, patient efficiency, the trapped nematode's own struggling movements apparently triggering the fungus to release additional adhesive and, eventually, digestive compounds directly at the point of contact, a remarkably targeted response from an organism with no nervous system whatsoever to coordinate it. It stands as one more quiet example of how thoroughly this whole patient kingdom continues to blur the neat categories we're used to sorting the living world into, decomposer and predator both, sometimes within the very same unassuming mushroom destined for tomorrow's dinner plate. High in the mountain forests of Japan, growing in a close, careful partnership with red pine roots, a mushroom with a dense, meaty texture and a distinctive, spicy aroma has been treasured for centuries as one of the most prized and expensive foraged foods in the entire country. This is matsutake, and like the truffle and the morel before her, she has never been successfully cultivated at any meaningful commercial scale, remaining instead a genuinely wild harvest, foraged carefully each autumn from very particular, carefully guarded patches of forest, sometimes passed down quietly within families across generations. Each patch's exact location treated with the same careful discretion a favorite fishing spot might receive anywhere else in the world. Matsutake holds a particularly significant place within Japanese culture too, traditionally exchanged as a genuinely meaningful autumn gift between family members and business associates alike, her considerable price reflecting both her rarity and her deep cultural association with the changing of the season. In recent decades, as domestic Japanese forests have produced smaller harvests than in generations past, careful supply chains have developed connecting matsutake forages across the Pacific Northwest of North America directly to markets in Japan, an entire international trade built around satisfying demand for one particular, deeply beloved mushroom. And scattered here and there across old, mossy logs and disturbed patches of soil, tiny mushrooms barely larger than a grain of rice form small clusters resembling miniature birds' nests, each one containing several even tinier, egg-shaped structures packed neatly inside. These are aptly named bird's nest fungi, and their strategy for spreading spores ranks among the most delightfully specific found anywhere in the fungal kingdom. A falling raindrop, striking directly into the tiny, open nest at just the right angle, splashes one of the small egg-like structures out and away, sometimes traveling more than a meter from the nest itself, trailing a fine, sticky thread that catches on nearby grass or twigs, anchoring the spore-packed egg exactly where it landed until it's eventually ready to release its own contents into the surrounding soil. Researchers studying the precise mechanics of this same splash cup dispersal have found the nest's own cup shape carefully optimized through evolution to direct a raindrop's impact with remarkable, reliable precision, launching the small egg at close to the ideal angle and speed needed to carry it clear of the parent nest and its own immediate, already well-colonized surroundings. It's a genuinely elegant piece of natural engineering, refined patiently across countless generations to make the very best possible use of nothing more than an ordinary falling raindrop. Rest here a while longer, surrounded by all these different careful strategies, prized delicacies, and startling predators and miniature raindrop-powered nests alike, each one simply another patient, quiet variation on the same underlying, ancient theme. This whole fungal kingdom has spent hundreds of millions of years refining. Let's drift now much further back, away from any single mushroom or forest at all, toward the very deep well of time this whole patient kingdom first emerged from. Fungi, in one form or another, are believed to have existed on this planet for well over 1 billion years, quietly present long before anything resembling a plant had ever taken root on dry land, long before the very first insect crawled across any surface, long before nearly anything else we might picture when we imagine ancient life on Earth. Fossil evidence suggests that fungi may have played a genuinely crucial role in helping plants first establish themselves on land at all, forming mycorrhizal partnerships with the very earliest land plants, simple, moss-like organisms with no true roots of their own, entirely dependent on a fungal partner to help draw water and minerals from the surrounding rock and soil. Without that same ancient partnership, some researchers argue, plants may never have successfully made the difficult transition from water onto dry land in the first place, meaning the entire, immense history of terrestrial plant life, and everything that followed from it, may ultimately trace back to a quiet, patient fungal partnership formed hundreds of millions of years before anything resembling a modern forest ever existed. It's a genuinely striking thought to sit with for a moment, that the tall trees, the soft moss, the fallen leaves scattered across the very forest floor we've spent this whole journey wandering through, may all ultimately trace their own existence back to that same quiet, patient fungal partnership, formed so long ago, between the very first tentative plants and the fungi patient enough to help carry them safely onto land. One especially puzzling fossil, found preserved in rock formations dating back roughly 400 million years, shows evidence of an organism called prototaxites, a strange, trunk-like structure that could grow to considerable height, taller than many of the plants living alongside it at the time. 4 years after its initial discovery, researchers debated exactly what kind of organism prototaxites actually was, some suggesting it might be an early plant, others an early form of lichen, before careful, patient chemical analysis of the fossil's own internal structure eventually confirmed it was, in fact, an enormous fungus, standing as one of the tallest organisms alive anywhere on the planet at that particular point in deep time, towering over the low, simple plants that had only just begun their own slow journey onto dry land. That same long scientific debate over prototaxites offers a genuinely fitting example of just how patient good science often needs to be. Nearly a century and a half passed between the fossil's initial discovery and the careful isotopic analysis finally confirming its fungal identity beyond reasonable doubt. Generations of researchers building patiently on each other's careful, incremental work, each one adding some small piece toward an answer none of them alone could have reached, until finally, slowly, the true picture came properly into view. Let your own thoughts settle into that same immense stretch of time for a moment, without needing to fully grasp its scale. There's nothing you need to calculate or measure precisely right now. Simply resting alongside a story this old is enough, all on its own. Fungi have also demonstrated a remarkable, quiet resilience across each of the great extinction events that have periodically reshaped life on this planet. Researchers examining the fossil record from the period immediately following the asteroid impact that ended the age of dinosaurs have found something genuinely striking. A sudden, dramatic spike in fungal spores preserved in the rock layers from that exact same moment, evidence that as so much other life struggled or disappeared entirely in the aftermath of that same catastrophic impact, fungi thrived, quietly and efficiently breaking down the sudden, enormous abundance of dead and dying plant material left behind across the whole planet. That same fossil spike, sometimes referred to by researchers simply as the fungal event, remains one of the clearest, most striking single pieces of evidence marking the exact geological moment the impact occurred, a thin, distinctive layer found in rock samples collected from locations scattered right across the globe, each one showing the very same sudden surge in fungal material at precisely the same point in the geological record. An entire planet's worth of quiet, patient fungal response, captured permanently in stone. That same pattern, fungi flourishing precisely when so much else struggles, appears to repeat itself across several other major extinction events preserved in the fossil record too, a quiet, consistent signature of exactly the same patient, opportunistic resilience we've circled back to again and again throughout tonight's journey. Where other forms of life depend on stability and favorable conditions to thrive, fungi seem to thrive precisely in the aftermath of disruption, patiently, efficiently converting whatever sudden abundance of decay and destruction happens to be available into the raw material for whatever comes next. It's worth sitting with that same quiet resilience for a moment, letting it settle gently alongside your own rest tonight. Whatever disruption or difficulty a day, a season, or an entire era might bring, this same patient kingdom has demonstrated, again and again across more than 1 billion years, that quiet, steady, unhurried continuation remains possible, even in the aftermath of the most dramatic upheaval imaginable. Let's return now to the warm, humid forest we visited earlier, back toward the world of the cordyceps and her patient, careful insect hosts, because fungi and insects share another kind of relationship too, considerably gentler and, in its own way, even more remarkable than anything we've discussed so far tonight. Deep within an underground chamber, carved out patiently over generations by a colony of leafcutter ants, something resembling a carefully tended garden grows quietly in the dark. Worker ants, having spent the day cutting small, precise pieces from leaves high in the surrounding canopy, carry each fragment carefully back down into the nest, not to eat directly at all, but to feed to a specific fungus the colony has cultivated deliberately across countless generations, an entire underground farm maintained with a level of careful, coordinated effort that genuinely rivals early human agriculture. The ants chew the leaf fragments into a fine paste, then carefully tend the resulting fungal garden, removing any competing mold or bacteria that might threaten the crop, and even producing their own natural antibiotic compounds, secreted from specialized glands, to help keep the fungus healthy and free from disease. In return, the fungus produces small, nutrient-rich swellings that the ants harvest directly as their primary food source, an entire self-sustaining agricultural system built and maintained by insects, refined patiently across an estimated 50 million years of shared evolutionary history between ant and fungus alike, long before anything resembling human agriculture ever began. Remarkably, when a young queen leaves her original colony to found a new one elsewhere, she carries a small sample of the colony's own cultivated fungus with her, tucked carefully in a special pouch, ensuring her own new colony can establish the very same fungal garden fresh, in an entirely new location, an unbroken agricultural lineage passed down directly from queen to queen across countless generations, considerably older and, in its own quiet way, considerably more established than any human farming tradition anywhere on the planet. Genetic studies comparing the cultivated fungus across many separate leafcutter colonies scattered widely apart have confirmed exactly this same unbroken lineage, finding the fungus grown in colonies located on opposite sides of a continent still closely related genetically, descended from that same original, patient partnership formed millions of years ago and carried forward ever since, queen by careful queen. An entire agricultural tradition maintained with a level of genetic continuity. Human farming, with its constant crossbreeding and deliberate genetic modification, has never come close to matching. Termites, in several regions of Africa and Asia, have developed a strikingly similar partnership entirely independently, certain species cultivating their own specific fungus within carefully constructed underground chambers, feeding it plant material gathered by worker termites and later consuming the fungus itself as their primary food source. That two entirely separate insect lineages, ants and termites, arrived independently at such a similar solution, deliberately farming a fungus for food rather than simply eating whatever plant material happens to be available directly, stands as one of the more striking examples of convergent evolution found anywhere in the natural world. Let your own thoughts rest gently on that same quiet, patient partnership for a moment, an entire hidden agricultural tradition unfolding underground, entirely out of sight, for tens of millions of years before anyone ever thought to look closely enough to notice it at all. Human agriculture, considerably younger though it remains by comparison, has developed its own long, careful relationship with fungi too, one stretching back thousands of years even before anyone understood the biology behind it. Long before Louis Pasteur's careful 19th century research finally explained precisely what yeast was and how fermentation actually worked, brewers and bakers across countless separate cultures had already learned, through patient generations of careful observation and repeated trial, exactly how to coax a reliable, consistent result from processes they couldn't yet fully explain. Mushroom cultivation itself has its own long, patient history too. Early Chinese cultivators are believed to have grown shiitake deliberately on prepared logs more than 1,000 years ago, carefully selecting logs that had previously produced a good harvest and using them to help inoculate fresh wood nearby, unknowingly practicing a form of selective breeding for productive fungal strains centuries before the underlying genetics were ever understood. But in mushrooms, among the most widely cultivated and consumed mushrooms anywhere in the world today, were first cultivated commercially in France, grown carefully in the cool, dark, abandoned limestone quarries beneath Paris, an environment that turned out, largely by accident, to offer close to ideal conditions for the mushroom's own patient, steady growth. Those same quarries beneath Paris continued to support commercial mushroom growing for well over a century, an entire underground industry operating quietly beneath the busy city streets above, workers descending daily into the same cool, dark tunnels first carved out to provide stone for the city's own buildings, now put to a gentler, unexpected second purpose entirely. Even today, the French word for this style of cultivated mushroom still carries a direct echo of that same underground origin, a small, lasting linguistic trace of exactly where and how this now familiar mushroom first learned to grow reliably under human care. Modern mushroom cultivation has grown into a genuinely significant global industry since then, an efficient, relatively low-impact way of producing a nutritious food source, often using agricultural waste products, straw, spent grain, sawdust from other industries, as the very substrate the mushrooms grow directly from, transforming what would otherwise simply be discarded into a fresh, useful harvest within a matter of weeks. Patient fungal mycelium once again quietly turning waste into something valuable. Exactly the same careful transformation it's been patiently performing in forests for hundreds of millions of years, now simply redirected, carefully, toward feeding people instead. Rest here a while amid this whole long, patient history, insects and humans alike, each finding their own particular way to work alongside this same ancient, endlessly adaptable kingdom, a partnership stretching back tens of millions of years on one side, and considerably more recent, though no less remarkable, on the other. Let's settle back into the quiet forest floor once more, back among the same patient mycelium threading invisibly through the soil beneath us, because researchers have recently discovered something genuinely startling about exactly how these fungal networks might be communicating within themselves, something that continues to reshape how carefully scientists think about the inner life of this whole ancient kingdom. Using extremely sensitive electrodes inserted directly into growing mycelium, researchers have detected regular patterns of electrical activity passing along the fungal threads, small, rhythmic spikes of voltage that bear a genuinely intriguing resemblance, in their basic pattern and timing, to the electrical signals nerve cells use to communicate within an animal's own nervous system. Some researchers studying this same activity have gone so far as to suggest these patterns might represent something resembling a simple form of language, entire sequences of electrical spikes that could, in principle, be grouped and analyzed the way a linguist might analyze the patterns within human speech. That same research found the frequency and grouping of these electrical spikes varied noticeably depending on what the mycelium happened to be doing at the time, different patterns appearing during active growth compared to periods of relative dormancy, and different patterns again when the mycelium encountered a food source or, in some experiments, physical damage to part of the network. Whether that same variation reflects anything resembling genuine communication, or simply a predictable, mechanical response to changing internal conditions, remains the central, actively debated question researchers continue working patiently to resolve. It's important to hold that same idea with appropriate care and patience, since this remains a genuinely new and actively contested area of research, and considerable scientific debate continues over how much meaning or intention to read into these same electrical patterns. Other researchers point out that similar rhythmic electrical activity can arise from simple, purely mechanical processes too, water and nutrients moving through the mycelium in a steady, pulsing rhythm, without necessarily implying anything resembling deliberate communication or thought behind it at all. The honest, careful answer, for now, is that nobody yet knows for certain exactly what these signals mean, or whether calling them a language captures something genuinely real, or simply borrows a familiar, evocative word for a pattern that may turn out to have a far simpler explanation once researchers understand it more fully. Either way, the underlying picture that's slowly emerging is a genuinely fascinating one. A vast, patient, underground network, subtly active in ways researchers have only just begun to properly detect and measure, quietly present beneath nearly every forest floor on the planet, doing whatever it does, entirely without needing our attention or understanding in order to continue. Let your own mind rest easy with all of that same open uncertainty for a moment. Not everything needs to be fully understood or explained right now, tonight, or ever. Some things can simply continue, quietly, patiently, exactly as they are, whether or not anyone watching fully understands the reasons behind them. Fungi have also learned to make a home in places that would seem, at first thought, entirely unsuitable for anything to grow at all. In the driest deserts, certain hardy fungal species survive for years in a completely dormant state, waiting patiently through however many seasons of drought it takes, before a single, brief rainfall finally arrives, triggering a sudden, short-lived burst of growth and reproduction completed within just a few precious days before the ground dries out once more and the whole patient cycle of waiting begins all over again. Researchers studying these same desert-adapted fungi have found their spores capable of remaining viable, ready to spring back into growth the moment conditions allow, for periods stretching well beyond a decade in some documented cases, an extraordinary feat of patient dormancy considerably longer than most seeds or spores from other kingdoms are able to reliably manage. It's a strategy built entirely around trusting that eventually, however long it takes, the right conditions will return, and simply being ready and waiting, quietly and without complaint, for exactly that moment to finally arrive. In the frozen ground of the high Arctic and Antarctic, other fungal species have adapted to thrive in conditions of near constant cold, their own cellular machinery specially adapted to continue functioning even at temperatures that would halt most other forms of life almost entirely. Researchers studying fungi recovered from deep within Antarctic ice, and even from the extreme, sun-scorched heat surrounding certain volcanic vents, continue to be surprised by just how widely tolerant this whole kingdom has proven itself across nearly every environmental extreme this planet has to offer. Even the wood inside an old, slowly decaying log can reveal a hidden fungal signature, if you know how to look for it. Certain fungi, as they patiently break down wood from the inside, leave behind fine, colored lines and patterns called sprawling, dark, branching borders marking the boundary between different competing fungal colonies quietly working through the very same piece of wood, each one releasing pigments as a kind of chemical fence, marking its own territory against its neighbors. Woodworkers and furniture makers have, for generations, prized this same spalted wood specifically for its beautiful, entirely natural patterning, an intricate, one-of-a-kind design created unintentionally by fungi simply going about their own patient, quiet business, entirely unaware that the resulting pattern might one day become someone's favorite tabletop. Rest here a while longer, considering just how many quiet corners of this world this same patient kingdom has managed to make its own, from the driest desert to the coldest ice to the inside of a single decaying log, never once needing recognition or understanding in order to continue its own steady, ancient work. Let's travel now to somewhere considerably less peaceful than the quiet forest we've spent so much of tonight wandering through, toward the interior walls of an abandoned nuclear reactor, sealed away and heavily contaminated with radiation following a serious accident decades ago. And yet, even here, in conditions that would be swiftly fatal to almost every other form of life, researchers exploring the site discovered something they never expected to find at all: colonies of dark, melanin-rich fungi growing steadily and calmly across the reactor's own damaged interior walls. Careful study of these same fungi revealed something genuinely remarkable. Rather than merely tolerating the intense radiation surrounding them, certain species appeared to be actively growing toward it, and further laboratory testing suggested the fungi might even be using the radiation itself as a usable source of energy, in a process researchers have taken to calling radiosynthesis, the same rich, dark melanin pigment that gives the fungi their color, apparently capable of absorbing radiation and converting at least some portion of that same energy into a form the fungus could use to fuel its own steady, patient growth, in a manner distantly reminiscent of the way a green plant's own pigment captures and converts ordinary sunlight instead. That same melanin pigment, researchers point out, serves other protective purposes across the wider fungal kingdom too, shielding many species from ordinary ultraviolet light in far gentler settings than an abandoned reactor, meaning this particular radiation processing ability may simply represent an extreme, patient extension of a much older, more familiar protective trait, adapted gradually to handle considerably harsher conditions than any of its relatives elsewhere ever needed to withstand. That same remarkable discovery has since drawn genuine interest from researchers exploring long-term human space travel, since a fungus capable of thriving on raw radiation, and of shielding whatever grows behind it in the process, might one day offer a genuinely useful, self-renewing form of radiation protection for astronauts on long journeys beyond the safety of Earth's own protective atmosphere. Samples of this same radiation-tolerant fungus have already been sent to the International Space Station for careful testing in the harsher radiation environment of low orbit, an entirely earnest, ongoing scientific inquiry into whether this same quiet, patient organism might one day help protect people on a journey toward Mars. Early results from that same orbital testing found a thin layer of the fungus capable of measurably reducing the radiation reaching instruments placed directly behind it, a modest but genuinely promising reduction that researchers believe could, with further careful refinement, meaningfully contribute to shielding astronauts or sensitive equipment during the kind of extended, deep space journey that would otherwise expose travelers to considerably more radiation than anyone experiences during a typical stay much closer to Earth. Fungi more broadly have proven themselves remarkably comfortable with the environment of space too. Several separate experiments carried out aboard the International Space Station have found that certain fungal spores not only survive the vacuum and radiation of low orbit, but in some cases grow even more vigorously once exposed to it, for reasons researchers are still working carefully to understand. It's one more quiet reminder that this whole patient kingdom seems to have anticipated, somewhere across its own immense evolutionary history, an extraordinarily wide range of conditions considerably harsher than anything most of us will ever personally need to endure. Let your own body rest easy here too, however strange or extreme this particular corner of the story might feel. There's nothing threatening in any of it right now, only the same quiet, patient resilience this whole kingdom has demonstrated again and again throughout tonight's journey, however unfamiliar the setting happens to be. It's worth pausing, too, simply to appreciate the sheer, almost overwhelming scale of the fungal kingdom as a whole. Researchers estimate that somewhere between 2 and 3 million distinct fungal species may exist across the planet, and yet, of that same enormous number, fewer than 150,000 have ever been formally described and named by science, meaning the overwhelming majority of fungal diversity on this planet remains, even now, genuinely unknown and unexplored, waiting quietly in forests, soils, and hidden corners of the world for someone patient enough to finally notice and properly describe it. New fungal species continue to be discovered and formally described at a genuinely steady pace even today, researchers regularly finding entirely new species hiding in plain sight, sometimes in thoroughly studied forests that seem, at first glance, unlikely to hold any real surprises left at all. Some newly described species turn out to have been growing quietly in city parks or even suburban gardens the entire time, simply overlooked amid the countless other, more familiar fungi sharing the very same patch of ground, a gentle reminder that genuine discovery doesn't always require traveling somewhere remote at all, sometimes only requiring the patience to look closely enough at what's already growing quietly nearby. Fungal spores themselves travel considerably further and more widely than most people would ever guess too. Researchers studying air samples collected from the upper atmosphere, several kilometers above the ground, have consistently found fungal spores present even at those same extreme heights, carried upward on rising air currents and capable of drifting for enormous distances, sometimes crossing entire oceans, before eventually settling back down to Earth somewhere entirely new. It's part of why certain fungal species manage to appear in such remarkably similar forms across continents separated by thousands of kilometers of open ocean, their spores having patiently, invisibly made that same impossible-seeming journey long before anyone was ever there to witness it directly. Some researchers studying this same atmospheric spore transport have even suggested that fungal spores drifting at extreme altitude might play a small, quiet role in weather formation itself, certain species carrying proteins on their outer surface capable of encouraging tiny water droplets to freeze around them, potentially contributing, in some small way, to the very process that eventually produces rain and snow. It remains an active, ongoing area of careful research, one more small, patient thread connecting this ancient kingdom to something as vast and as constant as the weather itself. Rest here a while longer, considering the sheer, quiet scale of everything we still don't yet know about this whole ancient kingdom, an entire hidden world of species still waiting patiently to be discovered, spores drifting even now somewhere far above us, carried by wind currents toward destinations neither they nor we could ever fully predict. Let's return once more to the open, grassy clearing we visited earlier tonight, where something else quietly reveals itself in the fading light. A near-perfect circle of darker, richer grass, scattered with small mushrooms rising up at regular intervals all along its curved edge. This is a fairy ring, and the genuine, patient science behind it turns out to be every bit as delightful as the folklore people once wove around it. A fairy ring forms as a single, roughly circular colony of underground mycelium spreads slowly outward from wherever its original spore first landed, growing at a steady, even rate in every direction at once, exactly the way ripples spread evenly outward from a stone dropped into still water. The mushrooms themselves only appear along the outer edge of that same expanding circle, where the mycelium is actively, freshly growing, while the grass directly above the ring often grows noticeably darker and more lush too, nourished by nutrients the mycelium has been quietly releasing into the soil as it patiently breaks down organic material underground. Some fairy rings have been growing for a genuinely long time. Researchers studying one particularly large ring found in France estimated it might be many hundreds of years old, its mycelium having spent centuries quietly expanding outward, a single patient circle slowly growing larger, year after unhurried year, long before anyone thought to measure or study it carefully. Aerial photography has, in more recent decades, allowed researchers to spot and measure fairy rings considerably more accurately than would ever be possible walking through the grass at ground level, entire fields sometimes revealing multiple overlapping rings of different ages and sizes once viewed clearly from above, each one a separate, patient colony expanding steadily outward at its own particular pace, some newly begun, others already decades or centuries into their own slow, circular journey. Long before any of that patient science was properly understood, though, Countless cultures across Europe had already woven their own explanations around these same mysterious circles, most involving fairies, elves, or witches dancing in a ring under cover of night, the mushrooms marking the very spot where their dancing feet had touched the ground. Old folklore often warned against stepping directly into a fairy ring at all, some traditions suggesting a person who wandered in might become trapped, forced to dance alongside the fairies until finally released, sometimes many years later, to find that far more time had passed in the outside world than they'd ever realized while dancing. Let your own thoughts rest easy in that same gentle, old folklore for a moment, without needing to sort fact from story too carefully right now. Both versions, the patient science and the old, dancing fairies, carry their own particular kind of quiet magic, each one simply describing the very same slow, expanding circle from a different angle entirely. In more recent decades, an entirely different kind of community has grown up around the patient, careful study of mushrooms too, considerably less magical in its language, though no less devoted in its own quiet way. Mycological societies, dedicated clubs and organizations focused specifically on the study and appreciation of fungi, meet regularly in cities and towns across the world, organizing group forays into local forests where members young and old wander together, carefully identifying whatever mushrooms happen to be fruiting that particular season, sharing knowledge patiently passed down from more experienced members to newer ones, exactly the way careful, hard-won expertise has always traveled. Best. In recent years, digital tools have joined this same old tradition too, smartphone apps and online databases allowing ordinary foragers anywhere in the world to photograph an unfamiliar mushroom and receive help identifying it from a wider community of more experienced enthusiasts, an entire informal, worldwide network of patient citizen scientists contributing, one careful photograph at a time, to a steadily growing collective understanding of where different fungal species grow, when they fruit, and how their populations might be shifting over time as the wider climate continues to change around them. Some of these same databases have grown large enough now to represent one of the more genuinely valuable resources available to professional researchers too, millions of carefully logged, geotagged observations building up a level of detailed, real-time coverage no single research team could ever hope to gather working alone. A professional mycologist studying how a particular species range might be shifting can now draw on many thousands of careful, patient observations contributed freely by ordinary enthusiasts, an entire generous, distributed research effort built quietly, one curious forest walk at a time. That same patient, collective effort echoes something we've returned to again and again throughout tonight's journey, an entire kingdom best understood not through any single dramatic discovery, but through countless small, careful, patient observations, gathered slowly, one quiet forest walk at a time, by generation after generation of people simply curious enough to look closely at what's growing quietly beneath their own feet. Rest here a while longer, amid all of that same patient, gathered curiosity, old folklore and modern careful science woven together, each one simply another way of paying quiet, careful attention to the very same patient, ancient kingdom. Let's move now toward the trunk of an old, standing beech tree, where a series of hard, woody shells extend outward from the bark in overlapping tiers, each one ringed with subtle bands of brown and gray, growing steadily larger year after year. Not unlike the growth rings hidden within the tree they're quietly, patiently feeding on. These are bracket fungi, sometimes called conchs, and unlike the soft, short-lived mushrooms we've met through most of tonight's journey, many bracket fungi are genuinely perennial, their tough, woody fruiting bodies persisting for years, sometimes decades, slowly adding a new layer of growth each season while the mycelium underneath continues its own patient work breaking down the tree from within. One particular kind of bracket fungus carries a history reaching back much further than most people would ever guess. When the extraordinarily well-preserved remains of a man who died more than 5,000 years ago were discovered frozen in the Alps, now known to researchers as Otzi the Iceman, he was found carrying two different types of bracket fungus among his careful, practical belongings. One, researchers believe, was likely used to help start and carry fire, its dense, fibrous interior capable of holding a smoldering ember for a genuinely long time once properly prepared, functioning almost like an ancient, entirely natural version of a matchbook. The other appears to have carried a more medicinal purpose, a species known even today to contain compounds with mild antibacterial properties, suggesting this same careful, prehistoric traveler may have understood and relied on fungi for practical, everyday purposes considerably more sophisticated than many people would ever assume possible for someone living so many thousands of years ago. Researchers examining exactly how Otzi's fire-starting fungus had been prepared found evidence it had been carefully dried and worked before being carried, suggesting a deliberate, practiced process passed down through his own community rather than any improvised, one-off solution. It's a small, quiet detail that transforms our picture of him from simply a lone traveler caught by misfortune into someone carrying forward genuine, inherited knowledge, exactly the same kind of careful, patient expertise about this ancient kingdom that mycological societies and careful foragers continue passing down between generations even today. Let your own thoughts rest gently on that same distant, careful traveler for a moment, carrying his own small, practical fungal toolkit across the mountains, trusting in exactly the same patient, quiet kingdom we've spent this whole journey exploring together. Elsewhere in this same forest, growing in soft, gelatinous, almost translucent masses along a fallen branch, a considerably stranger-looking fungus catches what little light remains. Sometimes called witch's butter for its bright, buttery yellow color and unusually soft, wobbling texture, this jelly fungus swells considerably after rainfall, absorbing water directly into its own soft tissue, before shrinking back down to a small, dry, barely noticeable husk again during drier stretches of weather, patiently repeating that same cycle of swelling and shrinking, sometimes for years, on the very same small patch of decaying wood. Old folklore across parts of Europe held that witch's butter appearing suddenly on a person's gate or doorpost signaled that a curse had been placed upon the household, and that the only way to lift it involved piercing the jelly-like fungus repeatedly with pins until it finally disappeared entirely. Considerably more mundane modern science explains the same mushroom's sudden, dramatic appearance as simply a natural response to recent rainfall, the fungus's dry, dormant form rapidly rehydrating and swelling visibly within just a few hours of a good, soaking rain, though there remains something undeniably charming in imagining an anxious homeowner, centuries ago, carefully pinning a small, wobbling lump of yellow jelly in hopes of protecting their family from harm. Mushrooms have also long been valued for a use having nothing at all to do with eating them, carefully harvested instead for the rich, varied colors that can be coaxed from their tissue through careful, patient dyeing. Certain bracket fungi and other wild mushroom species yield deep blues, warm oranges, soft pinks, and earthy browns when prepared correctly, an old textile tradition practiced across many different cultures, dyers learning through patient trial exactly which mushroom, prepared in exactly which way, would yield exactly which particular shade of color onto wool or other natural fiber. That same tradition continues quietly today among a smaller community of dedicated natural dyers, who gather specific mushroom species carefully and sustainably, simmering them slowly for hours to draw out their own particular pigment, transforming something that grew quietly from a fallen log or forest floor into a rich, lasting color worked patiently into cloth. One more example of this whole patient kingdom offering something valuable and beautiful to anyone willing to slow down enough to learn how to properly ask for it. One particular mushroom, favored especially among dedicated fungal dyers for the remarkably wide range of colors it can produce depending on subtle variations in preparation, has earned itself a small but genuinely devoted following within the natural dyeing community, entire regional dye guilds organizing group forays specifically timed around this same mushroom's own particular fruiting season, gathering carefully and sustainably from the same patch of forest, year after year, exactly the same patient, respectful relationship every other careful forager we've discussed tonight has learned to maintain with this same ancient, generous kingdom. Rest here a while longer, amid all of these different quiet gifts, fire starters and medicine, dye and old, gentle folklore, each one simply another small, patient offering from a kingdom that has never once asked for anything in return. Not every relationship between fungi and the wider living world unfolds quite so gently as the partnerships we've spent most of tonight exploring together. Let's pause for a moment beside the pale, weathered stump of what was once a towering American chestnut tree, a species that, within the span of only a few decades in the early 20th century, was reduced from one of the most abundant, important trees across the entire eastern half of North America to a scattered handful of struggling survivors, the devastating result of a fungal blight accidentally introduced from overseas, spreading through susceptible chestnut populations that had never encountered anything quite like it before and had no natural resistance built up to meet it. Researchers and dedicated volunteers have spent decades since working patiently to help this same beloved tree recover, carefully crossbreeding surviving American chestnuts with blight-resistant Asian relatives across many careful generations, slowly working to combine the resilience of one species with the particular growth habit and character people had always loved about the other. That same patient breeding program continues today, still many careful generations away from fully restoring the chestnut to anything resembling her former abundance, a genuinely long, unhurried project built on exactly the same quiet patience we've returned to again and again throughout the whole of tonight's journey. A broadly similar story unfolded with Dutch elm disease, another introduced fungal pathogen, this one spread from tree to tree by a small beetle carrying fungal spores on its own body, devastating elm populations across both Europe and North America across the 20th century, reshaping the visual character of countless towns and city streets that had once been lined with tall, graceful elms standing along nearly every avenue. Some cities responded to this same devastating spread with genuinely dedicated, patient conservation programs, carefully monitoring remaining healthy elms, promptly removing infected trees before the disease could spread further through the same connected root systems that make elms particularly vulnerable to rapid, underground transmission, and in certain cases, treating especially valued or historic trees directly with careful fungicide injections repeated every few years. A small number of these same carefully protected elms, some now well over a century old, continue standing today entirely because of decades of exactly this kind of patient, dedicated human care, a hopeful counterpoint to the otherwise largely difficult story surrounding this particular disease. These same difficult stories offer an important, honest counterbalance to so much of what we've celebrated together tonight. The very same patient, efficient decomposition that makes fungi so essential to a healthy forest can, when a particular fungal species finds itself introduced somewhere entirely new, without any of the natural checks and balances that kept it in careful balance back in its own original home, become instead a genuinely serious, disruptive force, reshaping entire landscapes in ways no one could have easily predicted or wanted. That same lesson has, in recent decades, shaped considerably more careful international regulation around the movement of live plants and soil between countries, quarantine inspections, and careful biosecurity protocols now standing as a direct, patient response to exactly the kind of accidental introduction that once devastated the American chestnut and countless elms across 2 entire continents. It's a genuinely practical, ongoing effort to extend the same careful respect we've shown this whole ancient kingdom throughout tonight's journey into concrete, preventative action, protecting not only the fungi themselves but the countless trees and forests that remain vulnerable to whatever new, accidental introduction might otherwise arrive next. Let your own thoughts rest gently with that same honest complexity for a moment. Nothing in the natural world, fungi included, is ever purely one thing or another. The same careful, patient qualities that make this kingdom so remarkable can, under the wrong circumstances, cause genuine harm too, a nuance worth holding alongside everything else we've learned together tonight, without needing to resolve it into any simpler, tidier story than it actually is. And yet, even from within this same complicated relationship between fungi and the wider world, one of the most transformative discoveries in the entire history of medicine emerged almost entirely by accident. In 1928, A researcher named Alexander Fleming returned to his laboratory after a short absence to find that a stray mold spore had drifted in and contaminated one of his bacterial cultures, and that a clear, bacteria-free ring had formed around the mold colony itself, as though the mold was somehow actively preventing the surrounding bacteria from growing at all. That same mold turned out to belong to the genus Penicillium, and the compound it was quietly producing, which Fleming named penicillin, would go on to become the very first true antibiotic, eventually saving what researchers estimate to be many tens of millions of lives across the decades since its discovery, an entire revolution in medicine sparked by nothing more than a single, ordinary mold spore drifting in through an open laboratory window at exactly the right, if entirely unplanned, moment. It took more than another decade after Fleming's own initial, accidental discovery before a separate team of researchers finally worked out how to purify and produce penicillin reliably enough for genuine, widespread medical use. A long, patient stretch of careful, unglamorous laboratory work standing quietly between one lucky accident and the medicine that would eventually transform surgery, childbirth, and the treatment of countless previously fatal infections around the world. Fleming himself remained genuinely humble about his own role in the whole story, often pointing out that nature herself had done the actual, difficult work of producing the compound in the first place, and that his own contribution amounted to little more than noticing, patiently and carefully, what was already quietly happening right in front of him. Long before Fleming's own careful, patient observation, though, various cultures across the world had already developed their own informal understanding that certain molds carried genuine medicinal value, applying moldy bread or other fungal preparations directly to wounds and traditional practices recorded across ancient Egypt, ancient Greece, and numerous other cultures scattered across history. An old, practical wisdom arrived at through patient, repeated observation long before anyone understood the careful biochemistry actually responsible for it working at all. That same genus of mold responsible for penicillin has offered up other quiet gifts too, considerably more culinary in nature. A closely related species is responsible for the distinctive blue-green veining and sharp, tangy flavor found in blue cheese, deliberately introduced into the cheese during its production and allowed to grow slowly through the curd over the course of weeks or months, transforming an ordinary block of cheese into something considerably more complex and distinctive. One more example of humanity learning, patiently and through considerable trial, to work carefully alongside this same ancient, endlessly useful kingdom. According to old, well-loved legend, blue cheese itself was first discovered entirely by accident too. A shepherd said to have left a piece of bread and cheese behind in a cool, damp cave, only to return weeks later and find it transformed by naturally occurring mold into something surprisingly delicious rather than simply spoiled. Whether or not that particular story reflects genuine historical fact, the same natural caves once used for this kind of accidental discovery continue to be used deliberately today by certain traditional cheesemakers. Their cool, stable temperature and naturally occurring mold spores considered an irreplaceable part of the cheese's own particular, patient character. Rest here a while longer, holding both sides of this same complicated relationship gently together. the genuine harm and the genuine gift, each one simply another true part of the same patient, endlessly varied story we've been following together throughout the whole of tonight's journey. Let's pause for a moment on something wonderfully strange about how fungi actually reproduce, a detail that quietly appends almost everything most of us assume about the whole idea of biological sex. While most animals, ourselves included, get by with simply two distinct sexes, certain fungi have evolved a considerably more elaborate system entirely their own. One particular species of mushroom studied closely by researchers has been found to carry something in the range of 23,000 distinct mating types, meaning any two individual fungal threads belonging to different mating types can potentially combine and reproduce together. An almost unimaginable degree of genetic flexibility compared to the simple binary most other organisms rely on. That same genetic complexity is controlled by two separate regions within the fungus's own genetic code, each one capable of existing in many different versions, and a successful pairing requires the two mating fungal threads to differ at both regions simultaneously, a system researchers have compared to needing two separate keys, each with thousands of possible variations, to match correctly before a lock will finally open. The sheer mathematical scale of possible combinations helps explain how a single fungal species can maintain such an enormous number of workable mating types while still ensuring enough genetic difference between any two successfully paired individuals to keep the resulting offspring healthy and genetically diverse. Researchers studying exactly why this same fungus evolved such an enormous number of mating types have suggested it may simply maximize the odds that any two fungal threads happening to cross paths in the soil will be compatible enough to combine successfully, an elegant, patient solution to the challenge of reproducing successfully while remaining essentially immobile, unable to actively search out a suitable partner the way a mobile animal could. Rather than needing to find one specific, matching partner among a simple pool of two possible types, a fungus with tens of thousands of mating types can pair successfully with almost any other compatible thread it happens to encounter by pure chance. Let your own thoughts rest easily with that same strange, wonderful complexity for a moment, without needing to fully untangle it right now. Some of nature's quietest arrangements turn out to be its most surprising, once you take the patient time to actually look closely. Fungi have also, on occasion, produced individual fruiting bodies of genuinely startling size, considerably beyond the modest mushrooms we usually picture scattered across an ordinary forest floor. Beyond the enormous puffballs we discussed earlier tonight, certain bracket fungi have been recorded growing to truly enormous proportions. One particular specimen discovered growing on a tree in China measured well over 10 meters across when its full, sprawling shelf-like structure was finally measured completely, likely representing many decades, if not centuries, of slow, patient, continuous growth from the very same fungal individual. Researchers who carefully documented that same record-breaking specimen noted it had likely been growing steadily, adding fresh tissue to its own outer edge year after year, since long before the surrounding forest had reached its current mature state, an entire silent witness to decades of change in the world immediately around it, never once needing to move, or hurry, or draw any particular attention to its own remarkable, patient scale. Similarly, enormous single mushroom caps have occasionally been recorded too. Certain chicken of the wood specimens, a bright orange, shelf-like bracket fungus prized by foragers for its meaty texture, found growing large enough to yield tens of kilograms of edible material from a single, continuous cluster, an entire abundant harvest from what began as a single, microscopic spore landing in exactly the right spot on a tree many years earlier. These same remarkable size records exist alongside something worth remembering from earlier tonight too, that any single, visible mushroom or bracket, however large it might grow, still represents only the smallest, most temporary portion of the far larger organism quietly persisting underground or within the wood itself. The true scale of any individual fungus considerably harder to measure directly than the impressive, visible fruiting body sitting in plain view above it. Rest here a while longer, considering both the smallest, quietest details of this whole kingdom, its remarkable, elaborate genetics, and its occasional, startling scale, each one simply another small facet of the same patient, endlessly surprising story we've been following together throughout the whole of tonight's journey. Let's drift now toward a quiet, traditional kitchen somewhere in East Asia, where shallow wooden trays of steamed rice rest covered beneath a light cloth, a fine, delicate dusting of pale spores settling gently across the grains. Within a day or two, that same rice will be entirely transformed, threaded through with soft, white mycelium and carrying a sweet, faintly nutty aroma entirely different from anything steamed rice would produce on its own. This is koji, cultivated from a specific mold species that has become genuinely foundational to an enormous portion of East Asian cuisine. Koji's own patient enzymatic work breaks down starches and proteins into simpler, more flavorful compounds, laying the essential groundwork for soy sauce, miso, sake, and rice vinegar alike, each one beginning with this same careful, controlled fungal fermentation before continuing on through weeks or months of further, equally patient transformation. Master brewers and fermenters across Japan, China, and Korea have refined the careful art of cultivating koji across many centuries, learning exactly which strain, grown under exactly which conditions, produces exactly the particular flavor a given soy sauce or miso tradition depends on, a whole quiet craft built almost entirely around trusting this same patient mold to do its careful work exactly right. Traditional koji makers describe the entire process as requiring constant, gentle attention during those first critical days, checking the rice regularly, adjusting temperature and humidity by hand, sometimes waking during the night to turn the rice and ensure the growing mold spreads evenly rather than concentrating unevenly in warmer patches. That same hands-on, patient vigilance, passed down carefully across generations of skilled brewers, remains, even in an age of considerably more precise industrial equipment, still valued and practiced by smaller, traditional producers who trust their own careful, accumulated experience over any purely mechanical shortcut. Not far away, in a kitchen tradition considerably further south, soaked, split soybeans are being laid carefully together, inoculated deliberately with the spores of an entirely different mold. Within a day of steady warmth, that same loose pile of beans has been bound together completely, transformed by a dense, white web of mycelium into a firm, sliceable cake known as tempeh, a traditional Indonesian food that has, in recent decades, found genuine popularity considerably beyond its own original home, valued for its firm texture and rich, nutty flavor. An entire dish built from nothing more than patient fungal growth binding humble soybeans into something considerably more than the sum of their parts. Traditional tempeh makers across Indonesia often wrap the inoculated beans in banana leaves rather than plastic, an old, practical choice that allows just the right amount of air to reach the growing mycelium while still holding everything together securely. One more small, patient detail refined across countless generations of careful, hands-on practice long before anyone thought to explain precisely why the banana leaf worked so reliably well for this same particular purpose. Let your own body settle a little further now, resting easy amid all of this quiet, careful transformation, trusting, the way generations of careful cooks and bros already have, that patient fungal work, given enough time and the right gentle conditions, reliably produces something worth waiting for. These same fermentation traditions share something important with nearly everything else we've explored together tonight, an entire, ancient collaboration between people and fungi, refined patiently across countless generations, long before the underlying biology was ever properly understood. A cook in ancient Japan preparing an early batch of miso, or a family in rural Indonesia binding soybeans into tempeh for the very first time, worked entirely from careful, observed experience, trusting a process they couldn't yet fully explain, exactly the way early bakers and bros elsewhere trusted their own careful relationship with yeast. That same trust continues today, carried forward by comparatively modern research finally explaining, in careful scientific detail, precisely why these traditional methods work as reliably and as well as they always have. Researchers studying koji's own enzymatic activity, for instance, have identified the specific compounds responsible for breaking down rice starch into the simpler sugars that give traditional sake its own particular depth of flavor, translating centuries of patient, hands-on craft into precise, modern chemical understanding, without ever once needing to replace the original, careful tradition itself. That same koji mold has, in recent years, also been formally recognized within Japan as a national fungus, a genuinely rare honor reflecting just how deeply this one patient, humble organism has woven itself into the country's own culinary and cultural identity across so many centuries of careful, shared use. Few other fungi anywhere in the world have earned quite that same level of formal, cultural recognition, a fitting tribute to a mold whose quiet, patient work touches an entire nation's daily table in ways most people never pause to fully consider. Rest here a while longer, amid the quiet, patient work of koji and tempeh alike, two more gentle examples of this same ancient kingdom offering exactly what's needed, slowly and reliably, to anyone patient enough to learn how to properly ask. Let's return once more to the quiet, mossy forest floor, and consider for a moment the deep, patient knowledge so many indigenous communities around the world have carried for countless generations regarding exactly which mushrooms grow where, and when, and how to gather them safely and sustainably. Across the Pacific Northwest of North America, indigenous communities have gathered wild mushrooms, including the same prized matsutake we discussed earlier tonight, for countless generations, developing careful, detailed knowledge about which specific forest conditions, which particular tree species, and which precise season would reliably produce the best harvest. That same knowledge, passed down carefully within families and communities long before any written record of it existed, continues to inform sustainable harvesting practices today. Careful techniques like cutting a mushroom cleanly at its base rather than pulling it up by the root, preserving the mycelium underground undisturbed so the same patch might continue producing a healthy harvest for many years, indeed generations, still to come. Elders within several of these same communities have spoken about mushroom gathering not simply as a practical, seasonal task, but as a genuinely reciprocal relationship with the forest itself, one requiring patience, respect, and a willingness to take only what's needed while leaving enough behind for the mycelium, and for future gatherers, to continue thriving undisturbed. That same careful philosophy, gratitude paired with genuine restraint, offers its own quiet, valuable lesson worth carrying forward into how anyone might choose to approach this whole patient kingdom going forward. Similar deep, patient traditions of fungal knowledge exist across countless other cultures too, from communities across Siberia and Scandinavia with their own careful understanding of the fly agaric we met earlier, to foraging traditions across parts of Mexico and Central America, where certain mushrooms have held ceremonial and medicinal significance within indigenous practice for many centuries. Knowledge considerably predating any modern scientific interest in the same species. In recent decades, modern medical researchers have, in fact, returned to examine certain of these same traditionally significant mushrooms with careful, renewed scientific attention, particularly regarding their potential role in supporting mental health treatment. Clinical trials conducted at several major research universities have studied, under carefully controlled medical supervision, whether certain compounds found in specific mushroom species might help support people navigating serious depression, anxiety related to terminal illness, or difficulty overcoming addiction, always administered within a carefully monitored clinical setting alongside professional therapeutic support, rather than anything resembling casual, unsupervised use. That same careful clinical framework typically involves extensive preparation with a trained therapist well before any session takes place, followed by the supervised session itself, and then several further sessions afterwards specifically focused on helping a participant process and integrate whatever came up during the experience, an entire structured, patient therapeutic process built around considerably more than the substance alone. Researchers running these same trials consistently emphasize that the careful therapeutic support surrounding each session appears every bit as important to the outcomes observed as the compound itself, a nuance easily lost in more casual public conversation about this same area of research. Early results from several of these same careful studies have been genuinely promising enough to prompt considerably larger follow-up research, and a few regions have begun cautiously exploring limited, carefully regulated frameworks for supervised therapeutic use as this same research continues to develop. It remains, appropriately, an area where careful, patient scientific study, conducted slowly and responsibly under proper medical guidance, continues taking priority over any hurried conclusions, precisely the same unhurried, careful patience that has defined nearly everything else we've explored together throughout tonight's whole journey. Let your own thoughts rest gently on all of that same accumulated wisdom for a moment, indigenous knowledge and modern careful research alike. Each one simply another patient way of learning to understand this same ancient kingdom a little more completely, one careful generation at a time. Beyond any of their medicinal or ceremonial significance, mushrooms have also developed their own quiet talent for camouflage and mimicry, several species evolving to closely resemble something else entirely as a means of either protection or advantage. Certain mushrooms growing low to the ground take on mottled, earth-tone colors and irregular shapes that blend remarkably well into the surrounding leaf litter, making them difficult for foraging animals to spot easily. Others, growing on decaying wood, develop textures and colors closely mimicking the bark or fungus already present nearby, an extra, subtle layer of disguise built directly into their own patient growth. Rest here a while longer, considering just how many quiet, careful strategies this whole patient kingdom has developed, some meant to attract attention entirely, like the fly agaric's bold red cap, others meant instead to avoid it altogether, each one simply another careful, patient answer to the very same basic question every living thing eventually has to answer for itself: how best to survive and quietly continue in whatever particular corner of the world it happens to call home. Let's pause for a moment to consider the careful, patient work of the researchers who first began mapping out the hidden mycorrhizal networks we discussed earlier tonight, since understanding just how that same knowledge slowly came together offers its own quiet lesson in patience. One forest ecologist, spending years of careful fieldwork tracing radioactive isotopes injected into individual trees, was able to follow exactly where those same marked nutrients traveled next, confirming for the first time with genuine scientific rigor that carbon and nutrients really were passing directly between separate trees through their shared fungal network, not merely theorized but actually, carefully measured and tracked. That same researcher has described her own early years of fieldwork as genuinely difficult and often discouraging, working largely alone through dense, difficult terrain, carrying heavy equipment for miles at a time, and facing considerable skepticism from parts of the wider scientific community before her own careful, patient data finally became convincing enough to shift broader thinking. It's a genuinely honest reminder that even research eventually recognized as groundbreaking often begins in relative obscurity, sustained mostly by nothing more than the researcher's own quiet, personal conviction that the patient work was worth continuing, regardless of how long it took anyone else to properly notice. That same body of research took years of patient, unglamorous fieldwork to build, walking slowly through forest after forest, carefully sampling soil, tracing fungal threads, testing one careful hypothesis after another, long before any of it produced the kind of clear, exciting finding that might make its way into a wider public conversation. It's a genuine reminder that so much of what we've explored together tonight, the Wood Wide Web, the mother trees, the careful chemical signals passing between neighboring trees, exists today only because someone, at some point, was willing to spend years patiently tracing threads too fine to see with the naked eye, trusting that the patient work would eventually reveal something worth knowing. Let your own patience rest easy alongside that same careful, unhurried research tradition for a moment. Whatever you're hoping to understand or resolve tonight doesn't need to happen quickly either. Some of the most valuable things anyone has ever learned took years of exactly this same quiet, careful attention to finally come into focus. Fungi have also become an unexpectedly useful, sensitive indicator of our planet's shifting climate. Researchers tracking mushroom fruiting patterns across many decades finding that numerous species now tend to fruit measurably earlier in the year, and in some cases considerably later into autumn too, than historical records from the same regions once showed. Long-running citizen science projects, some stretching back more than 50 years, built from the careful, patient observations of amateur mycologists recording exactly when and where mushrooms appeared each season, have become genuinely valuable scientific records precisely because of how long and how consistently they were maintained, offering researchers today a clear, detailed window into exactly how gradually shifting temperatures and rainfall patterns are already reshaping fungal behavior across entire regions. One particularly long-running record, maintained faithfully by generations of members belonging to a single mycological society, spans well over a century of careful, handwritten observations, an entire community's quiet, patient dedication to simply noting down what they found, when and where, season after season, without any way of knowing at the time just how genuinely valuable that same accumulated record would eventually become to researchers studying climate change many decades later. That same shift in fruiting patterns carries real consequences too, since so many other species, insects relying on a particular mushroom's spores at a very specific time of year, or animals timing their own foraging around a reliable mushroom season, depend on that same fungal timing remaining broadly predictable from one year to the next. Researchers continue watching these same shifting patterns carefully, one more thread in the immense, patient project of understanding exactly how a warming world is gradually reshaping the quiet, careful rhythms so much of life has depended on for longer than anyone can properly measure. Let's drift, too, toward water for a moment, since fungi have made a home there just as thoroughly as they have across every stretch of forest we've wandered through tonight. Marine fungi grow quietly along decaying wood and plant matter drifting through the ocean, and even within certain coral structures themselves, an entire branch of the fungal kingdom considerably less studied and understood than its more familiar forest relatives, simply because studying anything carefully underwater remains so much more difficult and resource-intensive than a simple, patient walk through dry woodland. Researchers who have managed to study marine fungi closely have found genuinely surprising diversity hiding within the ocean too, some species apparently unique to a single specific host, a particular kind of seaweed or coral, found nowhere else at all, echoing the same kind of specialized, patient partnership we've encountered again and again throughout tonight's journey, simply relocated entirely underwater, largely unexplored, waiting quietly for more careful, patient research to properly reveal just how much diversity remains hidden beneath the ocean's own surface. Freshwater fungi, too, quietly break down fallen leaves and branches accumulating at the bottom of streams and lakes, performing precisely the same essential, patient decomposition we've discussed all night, just carried out instead in a considerably wetter, less visible corner of the world. Rest here a while longer, letting your own attention drift easily between forest and water alike, trusting that this same patient kingdom continues its quiet work in every one of these settings, regardless of whether anyone happens to be watching closely enough to notice. Let's drift toward a shaded corner of the forest floor where a small, delicate orchid has managed to take root, because her own story turns out to depend on fungi even more completely and more delicately than almost anything else we've discussed together tonight. An orchid seed is remarkably, almost impossibly small, smaller than a grain of dust, carrying essentially no stored food reserves of her own at all, unlike the seeds of most other flowering plants, which typically pack a small, careful supply of nutrients alongside the embryo to fuel its very first stage of growth. Without any such reserve of her own, an orchid seed depends entirely on encountering exactly the right fungal partner in the soil the moment it lands, a specific mycorrhizal fungus willing to form an immediate, intimate partnership, effectively feeding the tiny, developing orchid directly until it's finally large enough to begin producing its own energy through photosynthesis. Without that same fungal partner arriving at precisely the right moment, an orchid seed will simply never germinate at all, regardless of how otherwise perfect the surrounding soil and light conditions might happen to be. That same dependency continues, for certain orchid species, well beyond simple germination too, some remaining significantly reliant on their fungal partner throughout their entire adult lives, quietly continuing to draw at least a portion of their nutrition through that same underground partnership rather than relying entirely on their own photosynthesis the way most other green plants eventually learn to do independently. A small number of especially unusual orchid species have taken that same dependency to its furthest possible extreme, losing their own green coloring entirely and relying completely on their fungal partner for nourishment throughout their whole lives. An orchid that has, in a very real sense, chosen patient partnership over independence altogether. That same complete dependency helps explain why orchids have historically proven so difficult to cultivate from seed directly, and why wild orchid populations can be so vulnerable to any disturbance of the surrounding soil and its resident fungal community. Remove or disrupt the specific fungus a particular orchid species depends on, and that same orchid may simply disappear from an area entirely, unable to reproduce successfully even if fully grown plants continue to survive for a time. Modern orchid cultivation eventually found its way around this same delicate requirement by growing seeds instead in a sterile nutrient gel that mimics the food a fungal partner would otherwise provide. But in the wild, this same intimate, essential fungal partnership remains every bit as necessary today as it's always been. Let your own thoughts rest gently on that same quiet, complete dependency for a moment, one small flower entrusting the entirety of her own earliest life to a patient fungal partner she has no way of finding deliberately, trusting instead that the right partner will simply happen to be there, waiting, exactly when and where she needs it most. In recent years, an entirely new and rapidly growing industry has begun exploring whether fungal mycelium, grown deliberately and quickly under carefully controlled conditions, might replace considerably less sustainable materials across a whole range of everyday products. We touched briefly earlier tonight on mycelium grown into sturdy packaging material, but researchers and designers have pushed this same idea considerably further since then, growing dense, tightly bound sheets of mycelium that can be treated and finished to closely resemble genuine leather, used already in a small but growing range of shoes, bags, and other goods. Unlike traditional leather, which requires raising an animal across a period of years before any material becomes available at all, a sheet of mycelium leather can be grown to usable thickness in a matter of days, feeding on nothing more than agricultural waste products that would otherwise simply be discarded. Researchers continue refining the exact growing conditions and finishing processes needed to make this same material even more durable and consistent, an entire promising new industry built from nothing more than patient fungal growth, guided carefully toward a genuinely useful, considerably more sustainable purpose. Several major fashion and design companies have begun quietly incorporating this same mycelium-based material into limited product lines in recent years, testing consumer response and durability under genuine, everyday conditions rather than only within a careful laboratory setting. Early feedback has generally been encouraging, though researchers and manufacturers alike acknowledge the material still has further refinement ahead before it can fully match traditional leather's own long-established durability and feel, a patient, ongoing process of gradual improvement not unlike so much else we've discussed together throughout tonight's journey. Rest here a while longer, considering both the smallest, most delicate partnership, a single orchid seed and her unseen fungal partner, and the newest, most forward-looking application, mycelium quietly grown into the shoes someone might wear tomorrow, each one simply another example of how thoroughly this same ancient, patient kingdom continues weaving itself into nearly every corner of the living world, old and new alike. Let's travel back now toward the cold, exposed landscape where we first met lichen earlier tonight, because in certain far northern regions, one particular kind of lichen plays a genuinely central role in an entire ecosystem's careful, seasonal rhythm. Reindeer lichen, spreading in soft, pale, branching mats across the frozen tundra, becomes the primary winter food source for enormous herds of reindeer and caribou, animals capable of digging carefully down through snow to reach the same patient, slow-growing lichen buried beneath. Because reindeer lichen grows so remarkably slowly, sometimes only a few millimeters across an entire year, an area of tundra that's been heavily grazed can take decades to fully recover before it's able to reliably support a healthy herd again. Indigenous reindeer herding communities across the Arctic have, for many generations, carefully managed their own herds' movement across the landscape with exactly this same slow, patient lichen growth in mind, rotating grazing areas deliberately to allow previously grazed patches the many years they need to properly recover, an entire careful, patient system of land management built directly around respecting fungal growth too slow for most of us to ever properly notice or appreciate on our own. That same careful rotation system has, in recent years, faced growing pressure from a warming Arctic climate, shifting weather patterns making some traditional grazing routes considerably less reliable than they once were, and researchers working alongside herding communities have begun documenting these same changes carefully, hoping to help preserve both the lichen itself and the many generations of accumulated, patient knowledge built up around managing it sustainably, even as the wider climate around it continues shifting in ways no amount of traditional experience could have fully anticipated. Lichen also reproduces in ways considerably more varied than the simple spore release we discussed with ordinary mushrooms earlier tonight. Beyond producing spores from the fungal partner alone, many lichens also release small, specialized fragments called sordia, tiny packets already containing both the fungal and photosynthetic partner bundled together, ready to establish an entirely new, fully formed lichen colony the moment they land somewhere suitable, without ever needing to wait for a spore to encounter a compatible algal partner separately, the way the very first lichen partnership must once have required. It's a considerably more efficient strategy for spreading, in its own way, a patient partnership already proven, sent out ready-made rather than needing to be reassembled fresh each time. A related reproductive structure called ascidia works on much the same basic principle: small, finger-like outgrowths that break away from the parent lichen's own surface, again carrying both partners bundled safely together, dispersed by nothing more than wind, rain, or the passing brush of a foraging animal. Between spores, sordia, and ascidia, lichen has assembled a genuinely varied toolkit for ensuring this same ancient, patient partnership continues successfully into each new generation, however slowly and cautiously that same expansion tends to unfold. Let your own body settle a little deeper now, resting easy, the way this same slow lichen rests undisturbed across the frozen tundra for years at a time, needing nothing more than patience to eventually thrive. Fungi have also quietly found their way into a considerable number of industrial processes most of us never think to connect with mushrooms at all. Specific fungal enzymes, carefully cultured and extracted in controlled laboratory conditions, are used widely in the production of certain cheeses, offering a reliable alternative to traditional animal-derived rennet, valued particularly by cheesemakers producing vegetarian varieties. Other fungal enzymes find their way into laundry detergent, helping break down protein and starch-based stains considerably more effectively at the lower washing temperatures many modern washing machines now favor for the sake of energy efficiency. Researchers have also spent considerable effort investigating whether certain fungal enzymes might help unlock more efficient methods of producing biofuel from plant waste material, the same patient. Efficient enzymatic breakdown fungi use naturally to decompose tough plant fiber in a forest repurposed instead to help break down agricultural waste into simpler compounds that can eventually be converted into usable fuel. Early research in this same area continues carefully, patiently, the way so much of the most valuable scientific progress always does, incremental and unhurried, one careful improvement building steadily on the last. That same quiet industrial presence extends even further than most people realize. Certain fungal enzymes also used in the production of fruit juice, helping break down cell walls efficiently enough to extract considerably more juice from the very same amount of fruit, and others used in the textile industry to help soften and finish denim fabric. An entire scattered collection of everyday products, food and clothing and fuel alike, quietly touched by this same ancient, patient kingdoms. Remarkable talent for careful, efficient breakdown. Rest here a while longer, considering just how many quiet corners of ordinary daily life, cheese and laundry and fuel alike, already depend, in ways most people never think to notice, on this same ancient, patient kingdom working steadily away somewhere just out of sight. Let's drift toward the edge of a quiet forest pond, where the surface sits perfectly still except for the occasional ripple of some small creature moving just beneath. Here, invisible to the naked eye entirely, some of the oldest and strangest fungi in the whole kingdom continue a patient life cycle unlike almost anything else we've explored together tonight. These are chytrid fungi, and they represent one of the earliest branches of the entire fungal family tree, diverging from all other fungi hundreds of millions of years ago and retaining, even now, a trait every other major fungal group eventually lost entirely: a genuinely mobile, swimming stage. A chytrid spore carries a single, whip-like tail called a flagellum, propelling itself actively through water in search of a suitable place to settle. A brief, active swimming journey unlike anything the spores of mushrooms, or yeast, or nearly any other fungus we've discussed tonight are able to manage on their own. Researchers studying chytrid fungi closely have found their swimming spores capable of sensing and moving toward chemical signals released by potential food sources nearby, actively navigating rather than simply drifting passively the way most other fungal spores do. A genuinely active, searching behavior that offers a small, fascinating glimpse of what the very earliest fungal ancestors, long before any of the more familiar mushrooms we've followed together tonight ever evolved, might once have looked like, swimming quietly through ancient waters hundreds of millions of years before anything resembling a modern forest ever existed. Most chytrids live quietly as decomposers within ponds, streams, and damp soil, breaking down organic material in freshwater environments the same patient way their forest relatives break down fallen leaves and branches on dry land. But one particular chytrid species has, in recent decades, become the subject of genuinely serious concern among researchers and conservationists, identified as the cause of a disease that has seriously affected amphibian populations, frogs and salamanders, across multiple continents. A sobering reminder that even this same ancient, mostly quiet lineage can, under the wrong circumstances, become a genuinely significant force of disruption too, echoing the same honest complexity we sat with earlier tonight regarding chestnut blight and Dutch elm disease. Let your own thoughts rest gently with that same complexity once more, without needing to resolve it into anything simpler. This whole patient kingdom holds both quiet, essential decomposition and, on rare occasion, genuine disruption, together, inseparably, the way almost everything true about the natural world eventually turns out to hold more than one thing at once. Let's turn now towards something gentler to close this part of our journey, back to the same quiet, delicate spore prints we discussed earlier tonight, because in recent years, that same simple identification technique has also become its own small, quiet art form. Enthusiasts carefully arrange a mushroom cap above paper or fabric, sometimes combining several different species with contrasting spore colors, deep blacks, soft pinks, warm browns, into a single, considered composition, allowing each mushroom to release its own natural pattern slowly overnight, producing a final image entirely created by the patient. Natural release of countless individual spores rather than any brush or pen at all. Some artists have taken this same idea further still, using spore prints as the basis for larger textile and paper works, or carefully preserving a particularly beautiful print through gentle fixative sprays, transforming what began as a simple scientific identification method into something closer to a quiet, patient collaboration between artist and mushroom alike. Neither one fully in control of exactly how the final pattern will turn out, both trusting the same unhurried, natural process to reveal something worth keeping. Consider both the strange, ancient chytrid swimming invisibly through still water, and the gentle, considered patience of a spore print quietly forming overnight, each one simply another quiet facet of this same endlessly varied kingdom we've spent so long wandering through together tonight. Let's travel now into complete darkness, deep within a limestone cave far below the forest floor we've spent so much of tonight wandering across, where sunlight has never once reached and never will. Even here, fungi have quietly made themselves at home, thin, pale colonies spreading slowly across damp cave walls, sustained not by any trace of light at all, but by whatever organic material happens to wash or drift in from the world above, fallen leaves carried in by underground streams, or the patient, accumulated droppings of bats roosting somewhere deeper in the same cave system. Cave-dwelling fungi have adapted in quiet, careful ways to this same permanent darkness, many losing the pigment that colors their forest-dwelling relatives entirely, since there's no advantage at all to producing pigment when no light ever reaches it to make any visible difference. Others have adjusted their own patient rhythms to align closely with the arrival of bat colonies, timing their own growth to take fullest advantage of the rich, nutrient-dense guano deposited beneath a roost, an entire careful, hidden partnership unfolding in absolute blackness, far removed from anything resembling the sunlit forest floor where this whole journey began. Researchers studying cave ecosystems closely have found certain fungal species so specifically adapted to this same guano-rich environment that they have found nowhere else on the planet at all, an entire specialized community existing only within the particular, narrow conditions a bat roost creates. Patient and thriving in total isolation from the wider fungal kingdom we've spent so much of tonight exploring together above ground. Let your own eyes rest easily in that same complete darkness for a moment, needing to see nothing at all right now, trusting instead in the quiet, patient sound of this same unhurried voice to carry you gently onward. Let's rise back up now, out of the cave and into the trunk of a stressed, weakened tree, where a tiny beetle, barely larger than a grain of rice, has bored a narrow tunnel directly into the wood. She carries with her, tucked into a specialized pouch on her own body, a small sample of a particular fungus, which she carefully introduces into the walls of her freshly excavated tunnel the moment she arrives. This is an ambrosia beetle, and much like the leafcutter ants we discussed earlier tonight, she doesn't actually eat the wood she's tunneled through at all, but rather cultivates this same carried fungus deliberately along the tunnel walls, feeding entirely on the fungus itself as it grows. That same fungus, in turn, depends entirely on the beetle for its own survival and spread, unable to travel between trees on its own, relying completely on being carried, deliberately and carefully, from one freshly bored tunnel to the next by a beetle who has, across millions of years of shared evolution, become utterly inseparable from her own particular fungal partner. Researchers studying different ambrosia beetle species have documented hundreds of separate fungal partnerships across the group as a whole, each beetle species carrying its own particular fungal cultivar in a specialized pouch called a mycelium, a small, dedicated storage structure that appears to have evolved independently several separate times across different beetle lineages, each one arriving, once again, at the very same basic solution to carrying a reliable food source safely along, wherever her own travels happen to take her. Researchers studying this same relationship have found many different beetle species, each one carrying and cultivating its own particular fungal partner, an entire hidden world of careful, coordinated agriculture playing out silently within the wood of countless trees, invisible unless you know to look for the fine, telltale tunnels marking a tree that's quietly hosting this same patient partnership. Rest here a while longer, considering both the fungus thriving in absolute cave darkness and the fungus carried carefully, deliberately, inside a tiny beetle's own body, each one simply another quiet example of just how many different, careful forms this same ancient partnership between fungi and the wider living world has managed to take, across every corner of this planet we've explored together tonight. Let's wander once more past a fallen, decaying log, where something genuinely strange rises from the buried wood beneath, dark, finger-like clusters pushing up through the soil in small, irregular groups, each one tapering to a rounded tip not unlike a set of pale knuckles reaching upward from below. Fittingly, and a little unsettlingly, this fungus carries the common name dead man's fingers, its dark, blackened, slightly swollen fruiting bodies persisting through much of the year on the very same patch of buried, decaying wood. Beneath the surface, dead man's fingers works through the wood using an unusually striking method, producing dark, thread-like lines that spread through the timber itself, sometimes called zone lines, remarkably similar in appearance to the sprawling patterns we discussed earlier tonight, though produced by this particular fungus's own especially distinctive, deeply pigmented growth. Woodworkers occasionally prize timber marked this way for its own dramatic, dark veining. One more example of a fungus feared slightly for its unsettling appearance, quietly producing something genuinely beautiful in the process of its own patient decomposition. Researchers studying these same zone lines have found they actually mark the defended boundary between two separate, genetically distinct fungal colonies competing for the very same piece of wood, each one releasing its own particular blend of dark pigment as a kind of chemical fence, holding its rival at bay along a shared, contested border. What looks, to the casual observer, like nothing more than an attractive pattern in a piece of old wood is really a permanent, visible record of a long, patient, entirely invisible contest, fought slowly over months or years between two competing colonies, neither one ever able to fully overtake the other. Elsewhere, in the cool, damp days of very early spring, small, cup-shaped fungi in a startling, vivid scarlet appear scattered across fallen twigs and damp leaf litter, easily one of the most visually striking mushrooms found in temperate forests anywhere, their bright red interior curling upward into a shape closely resembling a tiny, delicate bowl. These scarlet elf cups, appearing reliably each year at almost exactly the same early point in the season, have long served as one of the first quiet, welcome signs that winter is finally beginning to loosen its hold on the forest, a small, brilliant splash of color against an otherwise still largely bare and brown landscape. Naturalists keeping careful seasonal records across many decades have often noted the scarlet elf cup's reliable, early appearance as a genuinely useful marker of the changing season, her own particular fruiting window shifting gradually earlier in recent years across several well-documented regions, one more small, patient thread woven into the same wider pattern of shifting fungal timing we discussed earlier tonight regarding a warming climate's gradual effect on this whole ancient kingdom. Let your own body rest easy amid all these different, strange, and beautiful shapes, dark fingers and bright scarlet cups alike, each one simply another patient, careful answer this whole kingdom has developed to the same basic challenge of surviving and reproducing successfully in its own particular corner of the forest. Mushroom foraging itself has grown into a genuinely significant seasonal tradition across large parts of Europe, considerably more central to certain regional cultures than most people outside those same countries would ever guess. Across Italy, the annual porcini season draws enthusiastic foragers out into the forest each autumn, entire families making the same seasonal pilgrimage their own parents and grandparents once made, gathering the same prized, meaty mushroom that features so centrally in regional cooking traditions passed down across generations. Across much of Eastern Europe and Russia, wild mushroom foraging carries an even deeper cultural weight. Entire weekends during the autumn season traditionally devoted to family trips into the forest, baskets carried carefully home and sorted, cleaned, and prepared according to family recipes and preferences refined across countless generations. Certain markets across the region still sell wild foraged mushrooms sorted carefully by species, priced according to each variety's particular reputation and desirability, an entire seasonal economy built around nothing more than patient, careful searching through familiar, well-loved stretches of forest. Families across this same region often maintain their own closely guarded, favorite foraging spots, passed down carefully from parents to children across generations, the exact location treated with genuine discretion, not unlike the guarded matsutake patches we discussed earlier tonight. Children raised within this same tradition often describe their earliest, clearest childhood memories as walking quietly through the forest beside a parent or grandparent, learning patiently, one careful mushroom at a time, exactly which shapes and colors meant safety, and which meant it was time to simply walk on and keep looking. Rest here a while longer, considering all of these different traditions, Italian, Russian, and countless others besides, each one simply another quiet, patient way people across the world have learned to walk gently alongside this same ancient kingdom, gathering carefully from it, season after season, generation after generation, without ever quite exhausting its own patient, generous supply. Let's travel now toward a stretch of forest that burned some months ago, blackened trunks still standing amid the new, tentative green already beginning to push up through the ash-darkened soil below. Even here, in a landscape that looks, at first glance, thoroughly devastated, a particular group of fungi has quietly begun its own careful work of recovery. Certain species, known to researchers as pyrophilous fungi, meaning literally fire-loving, fruit almost exclusively in the specific aftermath of a forest fire, their dormant spores lying patiently in the soil for years, sometimes decades, waiting for exactly the particular chemical and temperature changes a fire produces before finally, suddenly springing into vigorous, abundant growth. Researchers studying exactly how these same spores manage to remain viable for such extraordinary stretches of time, patiently waiting through years or decades of otherwise unremarkable soil conditions, have found some species produce unusually thick, resistant spore walls specifically suited to long-term dormancy, an entire biological strategy built around trusting that the right, specific trigger will eventually arrive, however long the wait, and that patient readiness will always outperform any premature attempt at growth under conditions not yet properly suited to it. Researchers studying these same post-fire fungal communities have found they play a genuinely important role in a forest's own recovery, breaking down burned material efficiently and helping stabilize soil nutrients at precisely the moment they are needed most, before new plant growth has had any chance to properly re-establish itself. It's a remarkable, patient strategy, an entire fungal community built around waiting, sometimes for an entire human lifetime, for a single, specific, destructive event, before finally putting that same patient waiting to genuinely productive use. Let your own thoughts rest gently with that same quiet patience for a moment. Sometimes the most useful response to disruption isn't resistance at all, but simply patient readiness, waiting calmly for exactly the right moment to offer whatever help turns out to be needed. As our own understanding of fungi's essential ecological role has grown steadily more sophisticated in recent decades, conservation efforts have gradually begun extending the same careful protections long offered to endangered plants and animals to certain threatened fungal species too. Several countries have, in recent years, begun formally including fungi within their own official conservation assessments and protected species lists, recognizing that a rare, specialized mushroom depending on old-growth forest conditions can be every bit as vulnerable to habitat loss as any endangered animal sharing that same forest. This same shift represents a genuinely significant change in how conservation has traditionally been approached, since fungi were, for a very long time, simply overlooked entirely within most official conservation frameworks, protected only incidentally whenever the forests or habitats they depended on happened to also support some more obviously charismatic plant or animal species. One international conservation organization only formally added its first fungal species to its own global threatened species assessment relatively recently, a milestone that arrived considerably later than similar recognition for plants and animals, a quiet, honest reflection of just how long this whole patient kingdom has waited for the same careful attention other parts of the living world received considerably sooner. This same shift represents a genuinely significant change in how conservation has traditionally been approached, since fungi were, for a very long time, simply overlooked entirely within most official conservation frameworks, protected only incidentally whenever the forests or habitats they depended on happened to also support some more obviously charismatic plant or animal species. Researchers and conservationists specializing in fungi have spent considerable effort in recent decades making the careful, patient case that this same overlooked kingdom deserves direct, deliberate conservation attention entirely its own, rather than simply hoping it survives as an incidental side effect of protecting something else more visible instead. Rest here a while longer, considering both the patient fungi waiting years for exactly the right moment to help a burned forest recover, and the patient researchers working just as many years to help this whole overlooked kingdom finally receive the careful attention and protection it has always quietly deserved. Let's pause for a moment to consider something remarkable about exactly how a mushroom actually grows, a process that unfolds with such patient, steady force that it can, on occasion, push through surfaces most of us would never expect any living thing to overcome at all. Mushrooms have, in numerous documented cases, been found growing directly up through solid asphalt and even poured concrete, their soft, unassuming appearance belying a genuinely impressive amount of quiet, sustained pressure exerted from below. City maintenance crews in several regions have documented this same phenomenon repeatedly enough that certain persistent mushroom species have become genuinely familiar, if unwelcome, visitors along cracked sidewalks and parking lots, quietly reappearing season after season regardless of how many times the surface above is patched or repaired. The same patient mycelium continuing its own unhurried work entirely undisturbed somewhere beneath the surface the whole time. That same growth is powered largely by something researchers call turgor pressure, water drawn steadily into the cells at the growing tip of each fungal thread, building up internal pressure that pushes the tip steadily forward, cell by patient cell, in exactly the direction of least resistance. Rather than actively drilling or forcing its way through an obstacle, a growing mushroom instead patiently seeks out whatever tiny crack or weakness already exists in the surface above it, whether that's a hairline fissure in a slab of concrete or a small gap between paving stones, and simply continues pushing steadily against that same small weakness until it finally, gradually gives way. Researchers measuring this same turgor pressure directly have found certain fungi capable of generating pressure exceeding what a typical car tire holds, an extraordinary amount of quiet, sustained force generated by nothing more dramatic than water patiently accumulating within microscopic cells, entirely without any muscle, or lever, or mechanical advantage of any kind that would normally associate with genuinely powerful, forceful movement. It stands as one final, fitting reminder of just how much quiet, patient force can accumulate, cell by unhurried cell, given nothing more than time and the simple, steady willingness to keep gently pressing forward. This same patient, steady growth happens remarkably quickly too, once conditions are right. A mushroom's visible cap and stem, though they may seem to appear suddenly overnight, are actually built from cells that were already fully formed and simply compressed tightly within the underground mycelium beforehand, ready and waiting. When the right combination of moisture and temperature finally arrives, those same preformed cells rapidly absorb water and expand, causing the mushroom to seem to spring up almost magically over the course of just a few hours, when in truth, the actual, patient work of preparing that same mushroom was already quietly complete days or even weeks earlier, waiting only for the right moment to finally, visibly unfold. Time-lapse photography capturing this same rapid expansion has become a genuine favorite among nature documentary filmmakers, compressing what looks, to the naked eye, like sudden, almost startling growth into footage that reveals the actual, careful unfolding taking place cell by patient cell, each one simply expanding into a shape it had already, quietly, been holding in careful reserve. Let your own body settle into that same quiet image for a moment, everything already prepared and ready, waiting patiently only for the right conditions to finally, gently unfold, exactly the way your own rest tonight is already prepared and ready too, waiting only for you to simply allow it. That same patient preparation echoes something worth carrying with you as we finally begin turning back toward where the whole of tonight's journey first began. So much of what looks, from the outside, like a sudden, dramatic transformation is really the visible final step of a much longer, quieter process, prepared carefully and patiently, often for far longer than anyone watching from outside would ever guess. Let's return now, gently, to where we began. That same quiet forest floor, the same fallen log, the same small cluster of mushrooms that first caught our attention as the light was fading. The sky above the canopy has softened now, the deep black of full night giving way slowly to the faintest hint of gray along its furthest edge. The same soft mist still clings low to the ground, and the air still carries that same rich scent of old wood and damp earth we noticed right at the very start. Somewhere nearby, that same owl who called out once at the very beginning of our journey has long since gone quiet, and in her place, the first hesitant notes of a single, early bird drift softly through the still air, tentative and unhurried, the very first small sign that the whole forest is beginning, gently, to stir awake around us. The mycelium beneath this same log has continued its own patient work this entire time, entirely unaware of anything we've discussed together, entirely unaware that anyone was paying attention at all. It doesn't need our attention to continue. It never has. Long before either of us arrived here, and long after we've gone, this same quiet, patient network will keep doing exactly what it's always done. Breaking down what no longer serves the forest, feeding what still does, connecting root to root, tree to tree, in a partnership stretching back further than anything else alive on this planet today. Somewhere far from here, a truffle rests quietly beneath the soil, releasing her own patient scent into the earth around her. Somewhere else, a colony of leafcutter ants continues tending their own careful underground garden, exactly as they have for 50 million years. Somewhere in the permanent dark of a deep cave, pale fungal threads spread slowly across damp stone, needing no light at all to continue. And somewhere, quietly, a single spore, carried on some current of air none of us will ever trace, drifts toward wherever it happens to land next, entirely unconcerned with whether that landing comes today or several seasons from now. We've wandered a long way together through this same patient kingdom, across forest floor and underground network, through fire and flood and frozen tundra, into caves and clouds and the space station orbiting far above us all. We've met things beautiful and things dangerous, sometimes both at once. We've met a network older than most of the trees it quietly feeds, and a partnership so complete that one small flower entrusts the whole of her own beginning to it. We've met patient decomposers turning decay into new growth, and careful cultivators, ant and beetle and person alike, learning across countless generations exactly how to work gently alongside this same ancient, unhurried kingdom rather than against it. We've met bread rising slowly on a counter, and beer left open to whatever the night air happened to carry in. We've met a queen and carrying an entire agricultural tradition forward in a small, careful pouch, and a truffle dog trained patiently across many months to find something she'll never actually get to eat herself. We've met old folklore about dancing fairies and foxes, and careful modern researchers tracing radioactive isotopes through root systems for years at a time, each one, in its own way, simply trying to understand the very same quiet, patient kingdom a little more completely. And through every single one of them, we've returned again and again to the very same quiet truth, that patience, more than almost anything else, is what this whole kingdom has always understood most completely. Nothing here has ever needed to rush toward anything. Growth arrives when it's ready. Recovery arrives when it's ready. Even transformation, however sudden it might look from the outside, is really only the final, visible moment of something patient that's been quietly, carefully preparing itself all along. Let that same patience settle fully into you now too, wherever you happen to be resting. There is nothing left to understand more thoroughly tonight, nothing left to accomplish, nothing left to prepare for. Only this same soft, unhurried quiet, the same one that has held the whole of this long, gentle journey together from its very first quiet moment. Whatever small tension remains anywhere in your body can soften now, one last time, the way everything else tonight has softened, slowly, patiently, without ever needing to be hurried toward it. If any part of your mind is still holding on to some small detail from earlier, a particular mushroom's name, a particular shape or color, let that go too. None of it needs to be remembered right now. The forest doesn't ask its own mycelium to remember every single thread it's ever grown. It simply continues, patiently, trusting that whatever needs to remain will remain, and whatever doesn't was never essential to hold on to in the first place. The light continues to grow, gently, along the edge of the sky, the way it always eventually does. There's nothing more you need to do. Only rest, exactly as you already are, letting this same patient, ancient kingdom continue its own quiet work all around you, needing nothing from you now but the same soft stillness it's always been so quietly good at keeping company with. Wherever you are, whatever tomorrow holds, this same patient kingdom will still be there, working quietly beneath the surface, exactly as it always has, exactly as it always will, long after this particular story has finished being told. Rest now. Simply rest.